on challenges in global research in agriculture and technology. It's my pleasure to see you all who are joining today's session on this international webinar on challenges in global research in agriculture and technology, the day 2. We have with us our first lead speaker, Professor Ajay Rawat sir, who is from Lucknow, CSIR. And we have the principal of RBS College, Dr. UN Singh sir. We have the organizing secretary, Dr. P.K. Upadhyay sir, and head of the department Genetics and Plant Breeding. I would now request Dr. UN Singh sir, principal of RBS College, to please deliver the welcome address.
Hello.
Yes, sir.
Good morning, everyone. Good morning.
Good morning, sir.
Doctor.
It is my pleasure to see you all in this international webinar on challenging in global research in agriculture and technology. I, Dr. UN Singh, Principal RBS College Agra, and the webinar chairperson, extend my warm welcome to all the invited guests, delegates who are joining us, yet to join us on this virtual platform. Sir, I want to say that RBS College is the oldest college of India and it is the biggest college in Asia Pacific according to the area. I welcome the eminent speakers of today, day 2, Dr. A.K.S. Rawat, Executive Director, Ethno Medical Research Center, former head of the department, Pharmacologist and Anthropomycology Division, CSIR, NBRI, Lucknow. Dr. Sweta Thakur, Assistant Professor, SPU Simla University, Simla, and former Research Associate, ADR University, Malaysia, Pahang, Malaysia. Dr. Ulrich Berg, President, German Association of Homotherapy, Germany. Dr. Abdul Shokar bin Abad Razak, MD, UMP, EcoPest, SDN Faculty of CET, University, Malaysia, Pang, Malaysia. And Professor R.K. Kahar, Principal Scientist, IARI, USA, New Delhi, former DG and UPCR, Lucknow. Once again, I welcome all the delegates and eminent speakers on the behalf of RBS College. Thanks to everyone. Thanks to all the committee. Over to Dr. Upadhyay.
Thank you very much, sir, for your kind words. Now, I would request Dr. A.K.S. Rawat, sir, to please continue with his lecture for today. Over to Dr. Rawat.
Okay. Thank, thank you. Good morning to all of you, all dignitaries and all the speakers, learned speakers. I am Dr. Singh, Principal RBS College, Dr. Upadhyay, General Secretary of Society of Indian Society of Genetics and Biotechnology Research. At the outset, I am thanks to the Society and RBS College for inviting me for this important webinar, international webinar regarding the technology and its role in agriculture also globally, how we can go ahead. So, as we know, our three T that is a T for three T means information technology, agriculture technology, biotechnology technology. We have changed a lot in the last 3 to 4 decades, the socio-economic status of the people globally. Information technology has changed. Similarly, biotechnology has changed a lot. to improving the life of people's health sector and agriculture sector also. And regarding the role of genetics, we know the how genetics and the biotechnologies help in developing our production, agriculture production, I mean large green revolution also, and developing new variety, new breed, new uh, crops, variety of crops. They are for a high yielding, disease resistance, like that. But now we think not only that agriculture crops has only the things which give the farmers better income, but now as our government also and we all scientists also think now agriculture means should be integrated. We have to have agriculture crop along with we have to have a animal husbandry, poultry, metals plant, horticulture. So all these things together give better income generation to the farmer also and livelihood of the people. So we have to work on this line. So in present, I will just speak of medicine aromatic plant sector, how medicinal plant sector is very important for our healthcare, and especially in COVID area, post area during COVID pandemic, the herbs, where medicinal plant has play a very important role also. So, so how we can reach this sector globally also, how India can, I mean, go ahead with the competition, having a good variety, good product, herbal product. What are the lacuna? Why we are not up to that mark? Why our export is slow? So I will touch on that. So how CSR labs has worked on this area of our drugs. So just in coming few slides, I will tell you about the role of CSR. So that is a how CSR. The title of my presentation is. Uh, now you can have first slide, please. Hmm. That is CSI Initiative for Developing Standardized Scientifically Validated Herbal Products and the Future Strategies. Uh, globally, and what are the strategies we can have it? May I have first slide? Kindly focus it. Ansuman, first slide, please. Ansuman, hello, hello. First slide, please. Yeah. So the title of my presentation is what are the role of CSR? We have number of CSR institutes who are working in developing herbal products and how we have scientific development of this product. We have to make the more better well-rated product, her product for healthcare sector. So I will just give, I mean, what we can do in future also. Next one. So this is the, I mean, the aim of our CSR labs. Next one. Yeah. Next one. There are number of CSR labs who are working in the area of medicinal plant cultivation, developing new varieties, and product development, and the standard validations of the product so that we can have better health caring product also, as a medicine also, nutraceuticals, and the cosmeceutical also. Next one. So as you know, India, we have a number of different photography zones, and we have more than 17,000 flowering plants. And all these plants which give us number of medicine also. If you see, we have Indian system, Ayurvedic system medicine, United medicine system, homeopathy, Amchi, and the modern medicine also. So in these all three, four herbals in the Indian herbal health sector, we have more than 800 plants which are being used in different form. So in that way, India has blessed with the different vertical zones where uh, we can have grow these species. We can have plants species up to for high altitude Himalaya. to the deserts, we can even coastal area and whatnot. We have 12, more than 12 category zones where different crops with different medicine plants are grow. That is the strength of our country. Next one, and we have apart from that we have Indian system medicine. So this herbal sector is very important nowadays and a fast growing sector now. It has annual growth of 7 to 10%. You can understand. And if you see the global market is more than 5 trillion US dollar, and similarly China is in the top as for in other country also Germany, Europe, there are other countries, Europe also they are there, US also. But our India, we are, I mean, not done well. It is hardly 2.3 million US dollar. Although we have a, I mean, you can say more than 500 manufacturing units are there who make produce the herbal medicine. I mean, it may be Sira, it may be tablets. We can say Ayurvedic medicine also. We have the strength of that, and up to $1 billion US dollar worth of, I mean, dollar worth of medicine. I mean, we produce these products also in India. So, but only thing export is very low. That's the reason. So that's the seminar we are having today. How we can have global competition also. Where are the weaknesses? Although we have the number of more than 800 species, 8,000 species are known from this plant, and we have a good people, skilled people also. How we can reach that, increase our export also. So next one, next slide please. So what we have the opportunity in this century, India can do it. Although I already said, we have a rich bioresources, we have rich traditional knowledge about the plant, how one can use for their healthcare. We have different tribal medicine also. And apart from that, now for the last 2 3 decades, we have good scientific scientific experts are there, good scientific laboratories are there who are working. So only thing that we have a coordination, we have to have synergy, and we have convert these three in the to make the good herbal product, nutraceutical and also cosmeceutical. And this way we can give employment to our society, we can take care of healthcare also, and it gives the income generation to the farmer also. So, by cultivation of raw material, cultivar medicine crops, farmers can be rich. Later on, after same processing or processing, developing product, our industry will increase and our health sector will also benefit. So these things are very important, and our institute CSR, our government is also working this department in this way. And not only that, in the Ayush department is now is separate ministry, and this government have done a lot to understand the medicine plant have a great role. So these things we next one slide please. Yeah. So just about the CSR healthcare sector, drug development or drug pharmaceutical. So what CSR has done, I mean their contribution. If you see, we have the number of institutes who are working in the area of identification of plant species like India, it's a botanical medicine, and there are I am also Jammu. We have a Center Drug Research Laboratory. We have a CMAP also, Center Institute of Medicine Aromatic Plant, who work on the development new aromatic plants, varieties, and their processing, extraction. All these things are, I mean, in this area we are working on that. Whether it is single drug molecule, whether it is compound formulation. So we are working on this to make identify some molecules which are very important for the healthcare also, like artemisinin also. we have done it. There are number of things, Bako site, there are number of example I will give you one by one. So CSR work in this area, urban sector, and doing the chemistry of that, identifying the very biological compound which are biologically active, and then make the it as a after going development activity, going for scientific validation, a product. So these are the things we our CSR lab of next one. So these are the labs. I will not read all these things. It is available. As I told you, we have a center, the research laboratory. We have a CMAP, Indian Institute of Science in Jammu. So there are a number of our labs are there. So they work in different area, drug development to cultivation and development new variety of aromatic medicine. As you know, during this pandemic, COVID pandemic, we have highly demand of these medicine plants, whether it is a tulsi or simum or you can say lemongrass to making lemongrass oil, rose oil. There are a number of essential oils which are being used in the sanitizers being sanitizing, and also for the increased immunity of the people also. So these all we are working on last 30 years. Different labs are different places are working on this. So they have a very big role also. Not only that cultivation medicine plant, development medicine plant, drugs, or study validation of products, identify the single molecule as per the I.S.G.B.R.D guidance, as per the WHO guidance we are working and during this pandemic period also number of institutes are working in the testing of RT-PCR testing of COVID I mean this virus also and about developing new drug also. We know DRDO has developed D2O with collaboration with the CSR lab, I.S.G.B.R.D in Hyderabad. So that's why we have working in this area. Number of drugs already in the market. I will later on touch one by one. So these are the number of more than 10 to 15 labs are CSR labs which are situated in different parts of the country working in this area. So that our product can go, and accepted globally also. We can export our medicine which are scientifically validated because every medicine, everything nowadays has to have scientific validation as per the norms of the ICMR or the as per the norms of the our Drug Caution Act also. Next one, please. So I will just tell you the how all labs are working. The main philosophy of product development is that we have to show safety, efficacy, and quality. These things are very important. Whether we are going for the formulation, herbal form drug, or single molecule, we have to show safety first. It should be safe as per the guidance of ICMR or Drug Cosmetic Act also, and and you maintain the quality, which is very important. The quality of raw material, which variety you are growing, these things, all these things affect our product. So I will just touch one by one few points and give more time on the quality control of the drug and safety, how we have work out further. Next one. So after this next one, so thank you. So to have a genuine, authentic quality raw material for safety and its proper storage, so that we can have a right genuine raw material which can be go to the factory, our factory to produce the products. So how many things are involved? It is not one thing only. We have to have good agriculture practices for each crop. With how much fertilizer required, how to grow it, where to grow it, and when to harvest it. All good agriculture practices we have to follow it. Whether we have to go for the pesticide or better to go for the natural pesticide also, that things are required. All protocol we have developed it. Good collection if you are going to collect the raw material from the forest, how to collect it, how much you have to collect, so that for future we can conserve the plant species also, and proper identification of raw material. Whatever you are collecting should be properly identified. Similarly, with good ethical practices, good safety practices means the whatever material you have, whether you're going to crop, you have to see in future also we're going to test for the fungicide or pesticide residue, high metal residue, so all the microbial load also. So our product should be safe. I mean, it should have that quality the it should pass the limit of the all these I mean pesticide of fungicide microbial is there if there also. So these things, good storage practices, all these good things are required to get the best product. So it is integrated. There we have to have good team of scientific agriculture, good scientific biochemists, chemists also, and the pharmacist also, and marketing people so that their input go in way to I mean to educate farmer to collector also and the industry also how to have a best quality of a product. So first is raw material. If a raw material is up to the standardized and up to the without any I mean spurious things or without any pesticide, that is the main thing. Only this is the very important thing where our export sometimes India whatever they export sometimes they may they reported to have some I mean microbial load or pesticide in higher limit. So we have to take care of this. What are the means for the agriculture? This it require further discuss also. So I will just touch on the quality. Otherwise, in limited time we cannot go ahead. But only thing just I want to say you we have all the things documented by our National Management Board or our IT department or CSR labs. We have developed this. Even ICR also they have developed this protocol at Anand also. I mean, Director of Agriculture I mean, additional advertising medicine plan. D.B.R.D. in Anand also. Next one, please. So quality assurance and the installation, the I mean, I will focus is if you go to market, you can see the raw material how they are selling. I mean, one cannot say one can identify as a layman. You will say okay, but if you go scientifically, you may get high content of microbial also. Even the dust part is more. So these are things. So we have to know how to preserve it, how to keep it. This is a very important area for the to getting the right raw material, procurement raw material for the market. Next one, next one, please. So herbal drug installation of herbal medicine and their product is very important. So we have to take I mean, how we can identify how we can maintain the active constant of that bioreactive compound is that it should not be retarded or it should be have a deterioration should be not there. Whatever material we have, it should be within limit. We cannot control everything in our hand, but whatever if it there, so it should not go beyond the limit which is prescribed in our by the authorities for the safety point of view, I.C.B.R. or FDA or our Drug Control Act in our India also. So we have to take the. I mean, this is the main thing we are focusing even for our CSR lab. Next one. So this is what problem we have. If you see the market, if you go it, there is adulteration, substitution of same species. You can have, I mean, poor storage quality, which lead in the, I mean, development of microbial also, microbial load also. There are some species having two names. Then again, the confusion like Brahmi. In the name Brahmi, we have that two species. One is Asoka, one is Bacopa menori, other is the Centella asiatica. But if you go to market, you may get one. So, and the both chemistry different. Although action is same, but the chemistry different. So we have to know what species is being, I mean, marketed, which variety. That is important. We should go for the botanical name and the botanical name. Botanical name is a well accepted globally also. So we have to have this thing. But now, I mean, now people are slowly, we have, I mean, we are improving in the direction. There are good companies, good companies who are collecting raw material from the nature, also wild for a wild area like forest also, and properly storing, properly identifying to the method which is described the pharmacopoeia. So these are problem how to think also. Even seasonal variation is also important because India is a vast country. We have different vertical zones in India. We have arid zone also. Same time we have desert also. So sometimes same species grow in this area. So you will see the different chemistry also. So we have to see how much climatic variation is going on in the country and then come out with some, I mean, minimum, I mean, say standard for this plant species. That is a, I mean, work we are working and we have work on that in different species. I will just touch the one, two example of that. So chemical variation is very important. We should know which species growing best in which climate also, and how two species if there is an alternate, how you are going to identify. These things are very important to get the right botanically well accepted raw material. Next one. So quality assurance is very important, and now we can how we can identify right official species. We have developed monograph pharmacopoeia. We have in India as per the Cosmic Act 1940. Monograph is developed by I.S.G.B.R.D also, and in IUS we have Ayurvedic pharmacopoeia also, and our NBRI. We were when I was there, and still is going on. We have developed more than 100 I mean, monograph of medicine plant, which were later part of the pharmacopoeia also, Ayurvedic pharmacopoeia also. So these things now in public domain, we have now well said, I mean, having well documented pharmacopoeia, more than 800 plant species, standards are there, how you identify botanically, what is the name, what is the name, and everything about the monograph is there, how the plant is there. Its chemistry, its morphology, dosage, activity, everything is there. So we have to go for the monograph, which already India, in our country, we have developed this. So that's the first step, I mean, for our country, for last 20 years, so we have this monograph and pharmacopoeia of India also. Next one, please. So just I will give one two example of that. Why we sometimes fail, why our product fail sometimes when we collect the material from the nature or we cultivate also when we export it. The number is there also. Number example also. One is the chemotypes are there. India we have because we have different vertical zones and as we know the climate and the soil environment has an effect on the secondary metabolites. It varies place to place also. So we have to know how we can identify the best chemotypes so that our farmers can go ahead of the same species, same variety, same cultivar, elite cultivar, so that they will get the more better, I mean, cultivar crops and more biologic active raw material you get it from those crops. So, for example, this is one example of that. Different vertical zones are there. This is example tribulus terrestris. This is a plant tribulus where in India we can go through. So this plant will grow from, I mean, you can say from our cold desert to Ladakh to the Kanyakumari our in the and I mean South India to the desert also in Gangetic Plain also. So what we have done is we have collected more than more than 30 45 I mean sample we have collected different parts of that and we do the chemistry of that. As you know, tribulocin is the active compound which has number of biological activities, aphrodisiac, antioxidant also. So if next one, so these compound is vary place to place. So this is an example How you can see, I'm not going to detail of that because of time, but you can have an idea how the percentage of one active compound varies. Somewhere in arid zone, it is maximum. You can see the maximum,
Betul, betul.
0.600.
Apa ni,
And,
Syah, saya, you ingat tak yang dulu saya cakap abdomen locus tu?
and somewhere it is a minimum also. So I mean to say, only we get the high chemotypes from the desert area, that is our Rajasthan area, not in Indo-Gangetic plain, not in alpine. So we have to go, if you want to collect material, so we should go for the desert area, that is Rajasthan, Kutch, Gujarat area. It has a better, high content of biological active compound. So we to cultivate. If you want to cultivate, you can cultivate there also. That's why we have to make the study. One example also there, like Aculus calamus. It is a very important plant in our Ayurvedic system medicine. Uh, Aculus calamus is a it's rhizome being used in number of medicine and it
So
grow
sorry to
from
interrupt.
similar. Yeah, yeah, please.
Can we please sum up fast? We're running out of
Okay,
time.
cool. Okay. So I mean to say, the chemical variation, chemotic variation is very privileged in India also because it is a different particle zone. So we have to find right chemotypic chemotypes which can be go ahead for the better export of the medicine also, which is highly potential also. Next one. Apart from chemical variation, next one. So already I spoken. These agrochemicals we have collected from different parts of the country. Next one, and we identify the essential oil content. If you see, if you go next slide, please, the oil content varies. It is maximum 0.8% Himachal, while in UP area it is hardly 2.6%. Next slide, please. So it means there is variation in chemical content of the essential also. So we have to go to those area where we are getting high yielding variety of chemotypes to get the high essential oil content and try to grow same history in that area and document it. That we are working on that. Next one. And similarly, we have done like even identification right material. These are three species different from Bhuamla, Phyllanthus. Sorry, the captions are slow, but basically they look similar. But these three species are different. One is Phyllanthum amarus, one is Phytonus, Medapensis. But one can identify. Next one, we have through microscopic microscope you can identify these three species. If you go focus your under microscopy, you can see the margin, the clades, and this in the stomata size, stomata shape also vary. So macroscopically one can identify these species, and we have worked more powder more than 200 species like that. Next one, the role of microscopy is very important that way, and all these 200 now we have 800 plant species are there and they are all documented in our Ayurvedic form of India. So India has developed this. Ayush department has developed this pharmacopoeia, which is gadget official document, and one can have all these things about the morphological character by this. So we are a documented pharmacopoeia for the industry also. Next one, and we have contributed more than 200 species we have contributed. So similarly, we have go for the cultivation. Like we have developed, Shima have developed CMAP, our CSR lab, it developed one of the high content variety of khas also, vetiver, and yeah. And then we identify that, then we grow it, demonstrate, and give the sampling to the farmers of the UP or different parts of Papua. This is one of slide. We are giving some of the, products crops good variety to the vetiver, which is high content of essential oil. Next one. So CSR labs are working this area, developing new variety of valeriana, not only the lemongrass, and all these things. Uh, and these are, yeah. So in the nutshell, we can have products like I use product. We can go, we can like this Ayurvedic. This is the Brahmi, and directly you have right material, and you can directly make the formation also. And by the using going for the, you can go reverse pharmacology also, or modern formulation also. Reverse formulation is very important. We know the use of that plant. Only thing we have to demonstrate in the animal model and go for the safety, stability, and the is clinical trial, and you can go to market. Next one, like that we have directly you can make the powder extract of the material like Brahmi, Centella acetica, and you can make the product also. Next one. So I mean the CSI working there is not only Ayurvedic medicine, single drug also like artemisinin. Also we have a Google Lipid also. For Google Lipid we have number of drugs. Similarly we have the quality standard of Indian medicine plant, more than 800 plants. That all quality parameters are mentioned in our this these books also, which were developed under the I.S.G.B.R.D project also, and we have participated in it. So these are things are there. It means all the identification, right material, how to identify, we have documented, and now everybody, every person, every herbal industry can go ahead and for getting right is material also to comply this their standard. Next one. So that's what now we have been time because of time very short. So I would just sum up that in India we have Ayurvedic medicine which is well accepted for Ayurvedic system medicine, and there are phytomags which are only we can not export it. We can export it as a nutraceutical, but as a single molecules we have modern medicine. We have number like artemisinin. We have Agruculture also. So it is the market also. Centomin with CDRI, and more than 13 single molecule species also. And phytopharmaceuticals is a new drug of category which was notified in India in 2016, and it is extract bioactive extract which is being used for the different medicine. Next one. So these three categories are recognized in India also. That's I already told you we have to go forward way like pharmacology, reverse pharmacology also to whatever drug here, if we have knowledge about its use, directly we can go demonstrate in animals and do the chemical trial. Otherwise, the normal way is that identify compound and screen it. It's a long way to get the molecule. Best way is reverse pharmacology to get the standardized herbal medicine in a shorter period and efficacious product also. So the reverse pharmacology is the best way to develop a product for the short term. I mean, for the within 3 4 years you can have. And like modern molecule, yeah, it will take more than 20 years to get a single molecule, biologically active molecule, and safe molecule after doing clinical trial. Next one. Next. So these are the some guidelines. I will not go into detail. What are the parameter if you want to go the Ayurvedic medicine? So next one. So you have to we have a drug control act where we can go for to we have to like herbal drug development. We have to go for drug control act 1940 as per their rules. What are the rules are there? Next one. So what are rules there? We have to go. So that is important. Regarding that is knowledge. Yes, we have a database on that. She has ordered TKTL. We have it is different languages, more than three languages: English, Spanish, French, and one German also. And it is in public domain, and one can see how whatever plant species are being used by tribal with folklore. We have things now in documented and digitalized also. So that there are, I mean, we have CSR working on. Next one in Japanese, Spanish, French, yeah. So, like I was mentioning, we have the like Ashoka Surasaka. Yes, like directly one can make the extract and go as per drug content. Next one. So these are some examples. Next, this is a Brahmi. One can have different formulation. Can be powder. Can go as a tablet. It is a formulation. It is extract. It can be in capsule or tablet form. And we have done the different bioactivity of that. All the certificates. So these are products are in the market now. Only thing you can go for export of these. Next one, if you have to have better dosage form, like Pictureza could have very important plant now. Pictureliv is going to be a new drug from the CDRI. This is a plant for the liver, for the hepatic point of view, for liver disorder also. Hepatoprotective. So CSR labs are working in this area to get right extract of some medicine plant. Next one. So these are the guidelines I mentioned. I will not go into detail. Whenever a person want to develop some formulation, he had to refer Drug Control Act, which is the already gadget also, and accordingly you have to make the formulation and get the drug. I mean, pass through the Drug Control of India or state department. They have Ayurvedic drug controller. You have to submit your product dossier, and after evaluation by the committee, they give the approval. So these all revolutions are there in nutshell. Next one. So I will quickly. Time is being short. And these are some of the, I mean, different leads we have already said. I said, like in ICSR labs, the number of leads already in the pipeline for the anti-cancer drug, for hepatic protection drug, like that. Next one. So I will not going how we are developing. It is well known. If you want to extract, one can go for the extraction also and get the right material after doing the fractionation. Have a right material, one can go the different fraction and doing the animal study, the phase one, phase two, clinical trial. These all things are needed. For nutrition also we can do. Very good, fast growing sector nutraceutical. So in India is now also working the area nutraceutical. We have well framed guidelines for this and how important role is here for the we have now growing number of plants like crops which are nutritionally important, whether amaranthus, whether it is ashganda, whether it's I mean, ragi, everything, grains, I mean, coarse grain also, millets also. Now, if you have these things, you can make different formulation for breakfast also, for the nutrition. So this is the area we have to capture in future, nutraceutical from the from Indian side. When we know the terrace knowledge is there, you can grow right material, right crop, different climate, and make this product and give the nutrient value, safety also, and maintain this quality. These are things are very important. Next one. So you can go for the herbal product, nutraceutical for preparation, and after doing different phase one, phase two safety study and other clinical trial, one can have a product also. Next one. So like this, for example of similarly, we have CSR lab, CDRA lab now. They have developed number of products. So you can see two, three products are there because of I through mobile also. So different, but there are some product for the Google lipid is there. You know, for the brain also we have a Brahmi based from Bakupa we started extract there. Lipid lipid solubility point two. So we have the uh, Google lipid also. So CDR is working in number of products and for making the, I mean, her product to give the very little product for India, even for the export also. Next one, only few slides are there. Next one. So like that, CDRI, CMAP, NBRI, triple IM, they are all working in this the herbal drug area. And already for the liver, we have these all the coded extract and some are fractions. For the arthritis, Asperger, even cancer also we are working CDRI. So our CSR labs are working this. CDRI, Lucknow, Triple IM, Indian Institute of Integrative Medicine, Jammu also. So these are the active molecules which are, I think, within a few months, for years, I think they will be in the market. and it can be well accepted globally also. So next one. So if you have a herbal product doing scientific validation, identify right material, so it has forward linkage also, backward linkage also. Forward linkage is we can have good product like cost good product means like simple herbal product also, nutraceutical, cosmeceutical also. There's some example of those. We can have cough syrup having some few herbal material from that. So that next one even we can go for developing. We have already developed. These are our products which we have patented. Natural color and some of technology already in the market. This is cough serum. Next one. This is the our BGR 34 is one herbal medicine for diabetes. Apart from that, we have licensed patented this polyherbal your toothpaste also. We have made cough syrup, toothpaste, different things which are in the market now, in the market also. So I mean to say, we have to develop next one, please. So these products, this is a range of product NBR had developed lip balm, natural lip balm, which has natural color also. That was we are given to Himalayan company like BGR. These are things even CMF is developing number of pharmaceutical also. And CDRI and triple I.S.G.B.R.D, 4 5 labs. Jorad also we have one lab in Jorad also. They are all working there. They have to develop the herbal product pharmaceutical, nutraceutical. So that the things we have. Even phytopharmaceutical is a new category of drug which are working. Now this is the category of drug which are internationally accepted. So in better way you have a better extract, go for all activity as per the FDA or as per the ICMR guideline, and you can market it also globally also. So that is a phytopharmaceutical. We cannot go for Ayurvedic medicine. We cannot export as a medicine. It can be exported as a nutraceutical. So these are things in our different labs are working, and then that is the product phytopharmaceutical. Next one, please. So this was only in 2016. New category drug was was recognized in India. Next one. So we have to do go fast. These are parameters we have to follow to get the right pharmaceutical. We have to do seasonal variation, raw material, cumulative variation. We have to edit each SOP for the extraction. Next one, we have to do toxicity, safety, efficacy, all things parameters required. So next one. So we these drugs has just all parameters for development to follow which are in the modern medicine. You have to follow all the parameters of the modern medicine, single molecule, but it is extract form which are active and we can export it also as a medicine. Next one, huge market is there. So these are some of the different labs have developed different, leads are there. Next one, Ashgandha we are working, Mosul and Sirota we are working, cannabis also. So these are things are working. So this is this is the where I am presently in the Ithur Medical Center. It is in Northeast and Manipur, Hangmu. So we have developed some nutraceutical point of view, herbal tea, herbal, some nutritional drinks also, nutritional jam also, nutritional, next one, tea, some product for diabetes also we have identified. It is the my presently where I am working in the Manipur area. Next one. So herbal sector, we have the nutrition also. Soup also we have herbal soup also. That is one can go for the soup, one can have tea. We have developed some a good anti-helminthic formulation in the capsule form. Only thing we follow all the quality, safety, efficacy. These are the basic thing. And when we have developed four product, in a short of 3 years. And we have patented also. Only thing we have to go in the market now. We are going for identify some good company who can market this product. So there is a big scope to in India to get good raw material because we have different biological, I mean, sites and we have good laboratory also. Next one, so this is how we have, what are the concepts developed by food. What is herbal certificate is no agricultural, is no vitamin C, all the for the for foreigners we have collected, we have taken the some plants which are very available in our northeast area. So our study is like that. We have to work on the in the end to have a right raw material. For that, we have to right site, identify right area, and high yielding variety where agriculture, scientists, biochemists, biotechnologists, genetics, agronomists, they can develop new varieties of the medicinal plant which have high content of essential oil or high content of active compound. And later on, you can supply, store. You should have an idea now. All documentation is there. We have developed SOP for good storage, good extraction, and later on you go for scientifically validation of the product by different ways also. If you want to go making higher value medicine, it is a simple way. You have to demonstrate activity in animal model, and directly you can go. It's a clinical trial and its stability of doing the stability. Similarly, we can go pharmaceutical and single molecule also. It is long, so step. But now in India we have these facilities, CSR, and other also CS centers are there, I.S.G.B.R.D labs are there, other universities are there who are working in this area. So that in backlink, in the end, things are that we have to have backlinking with the farmer to give the better information about the variety, better agronomic practices, and finally you have the better income. I mean, the farmers can have better income while they're getting the good crop by supplying the genuine scientifically validated material to the company. Next one, I think this is the last one also. Next one, these are about all phytopharmaceutical. Phytopharmaceutical is a new category of that. So, so thank you. I think that's enough. I think. Yeah, next one. Thank you very much. I think I have taken, I think, a lot of time, and this is the in the end. I will just summarize that it is a medicinal plant in the area about. I mean, in addition to your traditional crop, rice, wheat, other crop, what you agriculture crop you are doing, it's a very important area, economic. I mean, a sector where a farmer can get better income and simultaneously, if you have a balanced soil, you can go for cultivation of aromatic plant. And only thing now we have different labs here, sir, which are going for developing different ICR lab also there who are developing new varieties of medicinal aromatic plant to get the lease, to get that to give the support to the farmer and with the good raw material to our good raw material to the industry also. So that's why your product is standing validated, it is safe, we can export it, and that's where we can have globally India can play it very important. And we have sure the future is bright in this area. Only thing we have to give more fine, we have to have farmers and and the other industries also to go for the better managerial practices to get the best. Thank you, sir. Thank you. Sorry for taking more time.
Thank you very much, sir. We have Dr. Ulrich Berg with us, who is the president of German Association of Homotherapy, Germany, with us. We have
Thank you,
Professor
Rosha.
Rajendra Kumar, sir, with us. Sir is the principal scientist of Division of Genetics and Plant Breeding at IARI and former Director General of UPCAR, Lucknow. Welcome, sir. We have Dr. Abdul Shoker bin Abud Razak sir with us from Malaysia. He is the Managing Director of UMP EcoPest, Malaysia. Good morning, sir. Welcome, sir. I'm extremely sorry, Dr. Berg, for letting you wait. as we were continuing with the session of
I'm not getting the sound.
with the session of Dr. Rawat. I'm extremely sorry for to all for letting you wait to enter the broadcast session. I guess if there are any questions, we invite questions. If anybody wants to ask something to Dr. Rawat, any questions to Dr. Rawat? Anyone?
There is no question. Other, I would like to compliment Dr. Rawat that this is the need of the time that we have to look for the traditional knowledge and their application and for the herbicides. Of course, there is one small query which is not related to science because Dr. Rawat was telling that now market people have to take these products. So my query is that institute is having business management unit or not, and if business management unit is there, what is the progress right now about your products?
Hello,
Please.
hello. Sound
Hello.
is not getting, sir. Just repeat the just question.
Yeah, my small, not question, other curiosity that I think your institute might
Volume is
be.
not. I'm not getting Ansuman. Volume.
So
I'm not getting the.
you're not able
Sorry.
to hear the voice.
Ansuman, you are able to hear me or not?
Yes, sir. I am able to hear. I am able
Okay,
to
that's
hear.
fine. So I think there is some problem with Dr. Ajay.
Yes. Okay, sir. No issues. Thank you for joining, Dr. Rawat. Thank you very much for
Yeah.
joining. Okay.
Sorry,
So,
I mean, I could not get I mean, the question he asked, but I certainly, if you mail me, I will reply for that.
No, no,
sure, sir.
Volume
that
is
Sure.
not.
that question.
Yeah.
Let him
Thank you.
know.
Thank you. Yeah.
Okay.
Sure,
You
sir.
can
Okay.
convey. Yeah. Yeah.
Yes, sir.
Thank
Thank
you,
you
sir.
very much,
Thank you.
Dr. Rawat, for your session. Thank you very much. Now I would request Dr. Ulrich Berg, who's the president of German Association of Homotherapy, Germany, to please continue with his lead lecture for today. And over to Dr. Berg now.
Okay, good morning. Actually, in India, good afternoon.
Very good afternoon, sir.
So,
We have your presentation on the screen, sir.
you have it. Okay, that's fine. But it doesn't work to change the slides. How do I do that? Okay,
It's working, sir. It's working.
yeah, now it's working. I had to do it in a different way. Okay, so my subject today is Agriculture and Homa farming, a sustainable solution for our planet. That's that's me and the contact informations. And nowadays, everybody is affected by this COVID-19, and which has led to a lot of suffering all over the planet. And in this situation, besides the suffering, also people have taken the chance to look inside and are now more concentrated on what is really important in our life. That is health, which also includes healthy food, healthy environment, then helping each other and building strong connections to family and friends. So whatever bad is happening, some good may also come out of it. Now, when we think of healthy food, healthy environment, we see that environmental deterioration and climate change are global problems. And what are the solutions? here in this presentation, I will show you one solution to purify our environment, air, water, soil, and produce healthy, nutritious food. And this suggested solution comes from Vedic knowledge in the form of homotherapy, and making use of traditional knowledge was suggested in the Convention of Biological Diversity meeting held by United Nations. Homotherapy with Agnihotra as its basic tool comes from Vedic knowledge and has wide-reaching beneficial effects on our whole environment. Our atmosphere, soil, water resources, and also biodiversity is increased. Nowadays, agrochemical farming has led to a severe pollution of the atmosphere, water resources, and the soil. Just look at soil health. According to data from FAO, the Food and Agriculture Organization of United Nations, we already have lost 1/3 of the world's top soil, productive soil. And they say, unless new approaches are adopted, the global amount of arable and productive land per person in 2050 will be only a quarter of the level in 1960 due to growing population and soil degradation. That, of course, will not be enough to feed the whole population. So we need some alternatives. Now, pollution. Let's look at the effects of pollution on plants. Of course, everybody knows the effects of pollution on humans. They are suffering a lot. If you live in Delhi, you know each winter you have so much air pollution that it's difficult to breathe. But similar things happen to plants also. If there's a lot of air pollution, the leaves are damaged, and that makes the plants more vulnerable to pests and diseases. Then photosynthesis is reduced, growth is reduced, and the yield goes down because of all these factors by 5 to 20%, according to recent studies. So we cannot continue with this normal chemical conventional form of agriculture, which is conventional, by the way, only since less than 100 years. In India, a little more than, say, 50 years. Before, organic farming was the conventional way of farming for thousands and thousands and thousands of years. Now we have to go back, and organic farm seems the only way to go. But regarding organic farming, there's an apprehension that when we move from chemical farming to organic farming, there will be a drop in yield. And this is why many farmers, they hesitate to do this step. But with organic farming, there will be no drop in yield, and yields will be even higher than with conventional farming. Just one slide I show, that was a series of studies, MSc studies at Dharwad Agricultural University in Karnataka. And they had examined yields and quality parameters in 4 crops: soybeans, cabbage, tomatoes, and okra. And you see, in all the 4 vegetables, there was an increase in yield, quite considerable in some cases. So yield is not a problem. With Homer farming, we can get at least the same yield like we get now. Now, what is this method which increases yield, reduces pests and diseases, and even improves health of the farmers? How does it work, and why is it so beneficial for growth of plants? The basic tool of home organic farming is Agnihotra, which is a simple fire done in a copper pyramid according to the biorhythm of sunrise and sunset. Comes from Vedic knowledge. And what is Agnihotra? That is the smallest and basic home healing fire, which is tuned to the specific biorhythm of sunrise and sunset. Here you see what you need. You need this small copper pyramid of fixed size and shape. You need some pieces of dried cow dung. Make sure it's pure cow dung, not mixed with buffalo or any other dung. You need pure cow's ghee, again, pure, not mixed with any other ghee. Then some rice grains, some Sanskrit mantras, which is exact vibration, and timetable of exact sunrise sunset, exact to the second. And what happens at sunrise? There's a description of sunrise in this ancient knowledge, which says that at sunrise, many fires, electricities, ethers, and more subtle energies come all the way from the sun to the earth. And this flood of energies has a subtle music in it. And the quintessential sound of that music is the mantra. And this explains why we have to be exact with both exact timing and second, the mantras, because the mantras are the vibration which exists at that time. So we have kind of resonance effect. Interesting to see this description of sunrise, which I got from an American astrophysicist, and he says at sunrise, million ampere currents head into the sun to the Earth at sunrise and out at sunset. And then what we do is the fire has to be fully ablaze at the time. We take a few grains of unbroken rice, akshata, and less polished rice, and then we apply some ghee to this rice. So every grain of rice should be covered by ghee. And there are two mantras, and we have two offerings of ghee. The mantras are in the morning, Suryaya Swaha, Suryaya Idam Namama, Prajapataye Swaha, Prajapataye Idam Namama. And after Swaha, you add one portion of rice mixed with ghee to the fire. In the evening, it is Agnaye Swaha, Agnaye Idam Namama, Prajapataye Swaha, Prajapataye Idam Namama. Then what happens is they said that a channel is created through all the atmosphere, and prana energy, life energy, which lies above the atmosphere, comes down through this channel. And an aura energy field is created around all plants nearby. So plants get nutrition, they get disease resistant. When the fire dies, the flame dies, then the energy comes back and is collected in the Agnihotra ash. This is why the ash can be used for making medicines, for applying to plants, for giving to the irrigation water to plants, and so on. Now, how does Agnihotra work? First, it purifies our environment, atmosphere, water resources, and soil, and thus brings all nature back to harmony. What are the effects of Agnihotra and Agnihotra ash on the environment? First, let us look at the atmosphere. Modern agricultural science mainly speaks of the effect of soil when they talk about nutrition for the plants. But according to ancient science, by the way, also according to biodynamic farming, more than 75% of nutrition to plants come from the atmosphere. And therefore, the state of the atmosphere is so important. Now, let us see the purification of atmosphere. Here we have biological contamination of atmosphere. You see Petri dishes were opened before Agnihotra, after Agnihotra. And you see the difference. Left side is before Agnihotra, right side is after Agnihotra, and you see a big reduction. That experiment was done a number of times and also published in different articles. So that's well established. But not only biological pollution, but also chemical and physical pollution are reduced by performing Agnihotra. Chemical pollution is mainly SOx and NOx. Physical pollution is particulate matter. Particulate matter, in the moment, a very important point, because these particles, the aerosols, also transmit the viruses, bacteria and viruses, which are now responsible for this COVID-19. So if you have less particles in the air, your risk of becoming contaminated, infected, is much less. This is why that is an important result. We see that chemical pollution, physical pollution, goes down by approximately 30 to 40%. And that's just by one Agnihotra. Of course, if you would do it regularly, this would continue the effect. Effect on water resources. There was one experiment done in Pune where raw water from a very polluted river became drinking water by just pouring the water through a column of Agnihotra ash. A glass cylinder was filled with Agnihotra ash, water was poured, and you see the difference. Before, it was very polluted, and all the parameters were improved. Just look at the biological pollution, standard plate count reduction of 93%, and most probable number went down by 98%. So at the end, we had drinking water according to the standards set up by WHO, by just pouring water through this filter of Agnihotra ash. Then another example, effect on bore well. That was from Australia. This farm, in Australia, this area is very dry compared to Rajasthan, and it's very dry. So having water is of utmost importance. These people drilled a bore well. They found water, which was lucky enough, but the water was highly saline and highly alkaline. pH was 9.5, salinity 1150 parts per million. This water was useless. Of course, you can't drink it. You can't give it to your animals. You can't give it to your plants. What they did is they performed Agnihotra next to the bore well and added Agnihotra ash once a week to the bore well. And after 6 months, pH went down to 9.2, nearly neutral, salinity to 720 parts per million. Now this water again was within the range of drinking water qualification, which WHO sets. So basically, their farm was saved by performing Agnihotra next to the bore well. There are many more experiments, but I can only show a few examples. Now, soil. Soil is of very, very important. And health of soil, only recently people talk about it more. Let us look at an old Sanskrit text more than 3,000 years ago. It said, "Upon this handful of soil, our survival depends. Husband it, and it will grow our food, our fuel, and our shelter, and surround it with beauty. Abuse it, and the soil will collapse and die, taking humanity with it." And we are close to this scenario. As I mentioned before, if we would continue like that in the year 2050, we would only have 25% of soil per person, which we had in 1960. So we really have to do that. What can we do? The soil has to be rejuvenated first by homotherapy. And in the rejuvenated soil, different types of microorganisms, starting from level of viruses, bacteria, fungi, algae, thrive. Thus, a healthy microflora and microfauna is created. Just look at one example, solubility of phosphate. We all know plants need phosphorus. That's one of the three main nutrients. You always have the formula NPK. And there is normally enough phosphorus in the soil, but it's there in a non-water soluble form, means plants cannot make use of it. This experiment done more than 30 years ago in US showed if you add Agnihotra ash to the soil, the percentage of water-soluble phosphate increased 3 to 4 times. And then the question, this experiment was confirmed recently in Germany, and they got the same result, increase in water-soluble phosphate in soil. Now the question was, how come? What is the mechanism? And there was a hypothesis that for this increase in water-soluble phosphorus is due to some phosphorus solubilizing bacteria. And that hypothesis has been confirmed in an experiment done in Ratnagiri a couple of years ago. They added Agnihotra ash to soil, and then they found that nitrogen fixing bacteria increased 100 times. And phosphorus solubilizing bacteria increased even 1,000 times. So that explains why water-soluble phosphorus increases if you add Agnihotra ash to soil. About other soil microbes, there were many experiments done, for example, in the I.C.B.R.D Institute, Central Institute of Subtropical Horticulture in Lucknow. And they, on one hand, examined pathogenic fungi, and they added Agnihotra ash, and there was no growth. But on the other hand, if it comes to beneficial microbes, then Agnihotra increased the growth of these beneficial bacteria. So that is very interesting. Another topic, sodic soil and acidic soil. There was one experiment done in district Unnao, which is between Lucknow and Kanpur. And the soil was very sodic, alkaline. pH was 9.86. And they kept 3 plots. One, they planted wheat with agrochemicals, normal. Then also wheat with vermicompost. Third plot, wheat with vermicompost and Agnihotra ash. And the pH of soil after one season only, you see here, with agrochemicals, it stayed the same, 9.86. Organic with vermicompost, it went down a little bit to 9.06, still quite sodic. But with Agnihotra ash, it went down by more than 2 points to 7.67. And, you know, with this pH, you can grow everything. Now you could think, okay, Agnihotra ash reduces the pH, but not only that. We had one example in Poland, a farm with highly acidic soil, pH was 4.4. Government agricultural engineer said nothing will grow there, forget about planting vegetables and so on. But people did, and of course they performed Agnihotra, added Agnihotra ash, and you see they grow everything here. Nobody of their neighbors can do that. They grow everything there. Later on, they also tested the soil pH, and pH went up to 7.2. So we see with Agnihotra and Agnihotra ash, we can improve both sodic and acidic soil. And in India alone, we have 7 million hectares of sodic and saline soil, and 2.5 million hectares we have alone in the Ganga plains of India. And then we have more than 6 million hectares of strongly acidic soil, pH less than 4.5, similar to the situation in Poland, especially in Arunachal Pradesh. And if these lands could be made productive again, that would be a great help, because that's together 13 million hectares. That's a large, large number. So what I suggest is to start a small project on either acidic or sodic soil or both, The investment for that is manageable, but the possible gain is huge considering all the 13 million hectares of sodic or acidic soil. Now, what is the effect of Agnihotra on plants? First, very simple experiments done in different places. That in even on school level you can do that. You take a petri dish, some tissue, add some seeds, and then you water. First is you water it with tap water. Second is you water with Agnihotra ash, but as any ash has some nutrients, that comparison would not be fair. You take any ash and the germination will be better. So what we always do is we also compare with control ash. Control ash is the ash which we get from burning the same substances, cow dung, ghee, some rice. But for control ash, we don't burn it in this Agnihotra pyramid, but in some other copper vessel, and not at the time of sunrise, sunset, and no mantras. But still, you see, there's a big difference between control ash and Agnihotra ash. Then we here you see the difference. Shoot length and root length were much better if you use Agnihotra ash. Then what is the effect on plants? That was a simple experiment which you can do just to see the effect. In any college you can do it, at home even you can do it. What are the experiences of farmers? That was mango in South America, where a farm was nearly dying. Then they started with Agnihotra techniques, homotherapy techniques, and you see in every year the yield went up to an incredible high number, and the quality was very good. That was mangoes in India, again in this Unnao area. In the middle, you see Dr. R.K. Pathak, who was at that time director of the Central Institute of Subtropical Horticulture, a mango expert. And here you see mangoes which produce so much that even from the stems, mango production was there. And the total production was 70 tons from 80 acres in the year 2008, which was an off year. And interesting, first, the neighboring farmers thought this guy is crazy, using Agnihotra and homotherapy methods. But then, all the farmers came and wanted to do the same thing because they had all the problems with different diseases and pests. And here on that farm, they were not there. This is tomato. You see at left side some fruit borer, tomato fruit borer, and they couldn't control it even with all heavy chemicals. And then they started Homa methods on the same farm, and within 3 months the problem was totally solved. Sugarcane. You see, this sugarcane plant was attacked by woolly aphid. It was a Homa farm, so the farmer was a little bit surprised. Why is this problem there? And then he checked and he found out that when they performed Agnihotra, the ghee which they used, they had bought from the market, and the ghee was not pure. It was mixed with buffalo ghee. And so immediately he made sure he gets good ghee. Also, he bought cows so that he makes his own ghee. And then, as soon as the proper Agnihotra started, 2 natural predators came and they took totally care of this woolly aphid problem. And on the same farm, he grew vanilla, and the vanillin content was so high that the lab, which tested the content, first thought they had made a mistake, so they had run the test again. because they had got 36% vanillin content by weight, and normal is 25 to maximum 28%. So also the quality increases, not just the quantity. Honeybee is very important in Homer atmosphere. They thrive, all over the planet. Bees are declining, but here in Homer atmosphere, very good. They do very good and also good production of honey. Now, effects of Agnihotra on plants. Now we saw some experiences of farmers, some controlled experiments which were done in different places, and that is, for example, flowers in a greenhouse in Uti, which is part of the Tamil Nadu Agricultural University. Then at Palampur Agriculture University, they did experiments with production of lemongrass and Tagetes minuta. Quality increased, oil content increased, and higher disease resistance. By the way, at the same place, they found that after they started Agnihotra, automatically some Brahma grew, which is a very powerful Ayurvedic herb. In the previous lecture, it was mentioned, and that came up. So naturally, nature restores. Then there were a controlled experiment at the University of Agricultural Science in Dharwad for MSc thesis. I already mentioned that in the beginning. Tomatoes, cabbage, soybeans, and okra were studied. And parameter studies were yield, pest and disease, quality, nutrient content. And in all parameters, we had an increase. Here you see yield. Then pests and diseases were decreased. Again, pests and diseases, a big reduction. Nutrient content of plants. That is one big problem. And, in the previous talk, it was mentioned that from medicinal plants, you could do some nice products like, soup or toothpaste, whatever. And but we have heard that the nutrient content of plants is reduced, and the medical power of plants also is reduced because of pollution. If you perform Agnihotra, this is a problem for Ayurvedic doctors also. If you perform Agnihotra on a farm, then all these parameters will be increased. Just one moment. Ich bin in einem Webinar. Ich rufe zurück. Which is very important. This slide we already have seen, increase in vanillin. Nutrient content in soybeans increases, cabbage, tomatoes, okra. All the different parameters of nutrition, they increase in Homer atmosphere. Effect on biodiversity. Biodiversity is a big problem now. Many people are not even aware of that. But for example, there was a study recently in Germany. They tested over 25 years the population of insects. And they found out within 25 years, the population of insects came down by 75%. That's incredible. Within only 25 years, 75% less. And then, of course, insects are very important. Just think of the bees and other insects for pollination. But also insects are food for birds, which are important also, and so on and so on. So that's a really big problem and shows how much biodiversity we have lost. And how does Agnihotra homotherapy help? Just two examples. These studies were done in Damnot, close to the Narmada River in MP, close to Indore. Uh, First is they checked the quantity of invertebrates in the riverbed of Narmada, and you see left side, I added up the numbers in these columns. Left side, these two columns show research stations upstream from the homotherapy place, which is shown in the middle, and then at the right side, two other stations are downstream, and you see there's a big, big, big difference. Much more of these invertebrates, much more biodiversity next to the Homer place. Similar studies they did with butterflies, number of butterflies at the Homer place, the largest number. And here you see one slide where you see all the colorful butterflies in this field of homotherapy, Goshala. So the end result is that if we implement Homer organic farming on a large scale, that will completely revolutionize the agricultural sector in India and all over the globe. And that will be a win-win situation for banks, which are suffering from bad loans, for farmers, for society in general, for every person. And in the long term, we get total eradication of poverty. So we can achieve both rural development and global food security. Now, just a few words about COVID, because that is affecting everybody. And Agnihotra can help in three different ways. First, it reduces the probability of getting infected. I mentioned these viruses attached to aerosols, and through aerosols, this COVID-19 is transmitted to other people. If there are less aerosols in the air, these viruses also, they settle down to the floor and they get inactivated. Number two is Agnihotra reduces pre-existing conditions like high blood pressure, heart problems, asthma, and so on, which normally lead to severe causes of COVID. Most people who die had some of these pre-existing conditions. But Agnihotra, you can reduce these, and thus you will have a less severe cause of COVID-19. And number three, very important, with virus, we don't have normal medicine like with bacteria. We have antibiotics. We don't have a similar medicine for virus infection. So most important is our immune system. So with Agnihotra, our immune system is strengthened. For example, we have less stress, less depression, less anxiety, and therefore the immune system is stronger, and it will be stronger to overcome an infection if you still get it. So that is, in short, the effect on COVID-19, how Agnihotra can help. So that's it. Here is my contact information, email, and here are some references. And that's it. Thank you. If you're interested, especially to do your own trials, your own experiments, please contact me, because that is ancient Vedic knowledge. And for our time, for our modern time, it is important that people understand the scientific aspects of it. So what I'm doing is I coordinate research on different aspects of this Agnihotra. And I'm happy if you want to be part of this effort, if you can contact me, and then we can see what you can do. There are some very simple experiments. There are some more experiments going deeper. A lot of interesting stuff can be done in this area. A lot of interesting subjects also for students who want to do their MSc or PhD or BSc thesis. Okay, thank you.
Thank you very much, Dr. Burke. Let me check if we have any queries from our audience and delegates to ask you. Um,
Yeah,
okay.
Dr. Ulrich, good afternoon.
Hi, good afternoon.
Yeah, I heard your lecture last time also, and this time also, and I have been hearing many of the lectures on Huma and impressed also. But as a scientist, one point always pinches me that it does not accommodate the statistical analysis. What is the exact experiment size, population size, and about your CD or SE, all those things. Without all these supports, science to what extent is going to accept? That's a question from my side. Of course, because there in one of your slide, I saw that it was showing 21% higher yield, and in case of others, it was somewhere 10% or like that. So, does it have been tested statistically?
See, not all the experiments which I've shown are gone through statistical examination, but some have some of the scientists have done that. The other experiments, you could say, these are more like pilot studies. But for example, we have done one project with HIV positive children, and there we have done the statistics, and it was showed a very significant difference. And some of these studies in the field of agriculture, like this MSc thesis, there also statistical analysis has been done. So if you want, I can you give me your email address, and I can send you the full studies.
Okay, Rajendra K64.
Oh, no, no, please. When I note it down, I will make mistakes. Either best you send me an email, uh,
Okay.
then
Uh,
uh,
I will request organizers to send my email to you because
okay,
I don't
fine.
have your email. But
Okay.
anyway, just I am typing on this chat box. You can note it.
Here you see my email. Do you see it?
Where I have
Oh,
not seen. Of course, it was in your slide, one of the
oh,
slide, but I could not.
the slide is no longer on. Okay.
Yeah, yeah. Now I have said no issue. Okay.
Okay, so please
Yeah,
send me an email and I will answer the question. Of course,
that's fine.
uh, we are still at the beginning of all the scientific studies. So many more things can be done, should be done. This is why I invite all the scientists to take part in that effort, so that we have a very broad set of results, of course, done according to all the scientific standards.
Yeah, that I understand, but primarily I am convinced with the therapy because it's not new, very new. Rather, it's one of the organic farming approach.
Yeah,
Uh, it can be, but it needs some scientific data and support so people accept it.
sure,
That's a problem. Yeah,
sure,
that's what I'm telling. Because
sure. And this,
everywhere, even I heard Dr. R.K. Pathak also.
yeah,
So there is always problem unless until you substantiate your data with the proper statistical tools. Nobody is going to accept you.
yeah. This is why why I give such talks and
Yeah, yeah,
invite scientists to jump on the bandwagon and help us. Uh,
yeah.
they're not
Actually,
enough yet.
no, I'm not against her. Actually, this is the need of the time. It should be done because our natural resources are consuming every day. Lot
Mhm.
of problem is going to take place in future, and we have to look for some alternates. So certainly, it's have potential for in the future, but it has to be. popularized and accepted at the public domain. Okay, thank
Sure,
you.
sure. Thank you.
Yeah. Okay, Manish.
Sir, we have a question coming for you. I personally didn't understood the question, but somehow I hope that you will be able to answer it. And the question is from Miss Pooja Singh, and she's asking, Sir, hormonal injection in crops and fruits used in India to rise the production, how to bar them, which are injections which are injurious to humans? Can Agnihotra or homotherapy help with it?
Um,
I hope you
see,
got the question.
maybe. Of course, this um, treatment of plants with hormones uh, can be very bad for our health because these hormones affect us also, and hormones they regulate a lot of problems in the physiology of the body. So better to avoid that. But there was an interesting study in Pune which showed that these uh, hormones, plant growing hormones, increase if you add Agnihotra ash and you perform Agnihotra. So the plant itself produces more of these hormones, and thus the growth is increased. And then we don't need to add these hormones from outside. Nature itself does it.
Okay, thank you very much, sir. I check if we can. Let me check if we can have a few more questions, please. Uh, Anybody who is interested to ask any question to Dr. Burke?
Maybe
I
best
don't
type
see.
in the chat box. But if there are no questions, okay.
Yeah, there's one thing, sir. I have a lot many questions, but I'm unable to ask them here one by one. Can I please get the golden opportunity to have a conversation with Dr. Burke through telephone? A person here wants to have you have a telephonic conversation with you as he's having a good number of questions to ask you.
Yeah, okay. He should give me his telephone number, and then I would call him.
Okay, so you got a golden opportunity which you asked for, and for sure, Dr. Burke will have a conversation with you. Uh, Very good. That's good for you. Thank you, sir. Thank you very much. No more questions coming up for you. That was a very clear, crisp, and clear presentation from your side. And now I would like to thank you. Thanks a lot, Dr. Burke, for your kind presentation and your kind words. Thank you very much,
Okay.
sir.
Thank you. Bye
Now
bye.
I would like to. Bye, sir. Thank you, sir. Now, I would like to invite Dr. Abdul Shokar bin Abdul Razak, sir, who is the Managing Director of UMP EcoPest, SDN BHD, Malaysia, and Faculty of CET, University Malaysia, Pahang, Malaysia. Over to you, sir.
Okay, thank you very much, Dr. Ashman. Okay, and hi everybody. Okay, Assalamualaikum warahmatullahi taala wabarakatuh, and very good afternoon to all of you. First, I will share my. You hear my voice, Ne? Clear voice?
Yes, sir. Your voice is coming perfect. It's
All right, thank you very much. Okay, now I share my slide to all of you. Is it appear in the screen?
Yes, sir.
All right,
Blockers
thank you
from
very
green
much.
to anti-pest for locust control execution plan using integrated of Moringa oleifera, Cerbera, odolum and high frequency supersonic.
Thank you very much, Dr. Shuman. Okay, first of all, I'd like to introduce myself. My name Abdul Shukur bin Abdul Razak from UMP Eco Personnel Bahad. So I'm the spin managing director of the company called UMP Eco Personnel Bahad, which means one of the spin-off company under University Malaysia Pahang. So I'm also a lecturer, a senior lecturer in Faculty of Civil Engineering and Technology, UMP. So today I would like to share to all of you, okay, regarding my product, Plockers. Plockers stand for prevention lockers. So from green to anti-pest for locust control execution plan using integrated of Moringa oleifera, Cabra odolum, and high frequency supersonic. Okay, the background study is this is the one of the system integrated, which means locust problem, especially in the Middle East. For your info, I'm going already went to Saudi Arabia during pandemic COVID-19 last month in June from what call sorry May and June from 20 May 22 to 18 June. So it's about almost 1 month. So regarding the collaboration between both countries about for eliminating locusts in Middle East. So I'm trying to use the green technology solution, which means we integrate the what called the plant called moringa. I'm sure that everybody know about the moringa oleifera, or in Malaysia called local name called kelo tree, and also the Cabra odolum, one of the plant tree for eliminating rain. So I'm trying to integrate all these plant and with what called the high frequency ultrasound, supersonic ultrasound. So which mean the supersonic frequency travel a speed greater than the speed of sound, and it can eliminate the locusts in that specific area. So this is a problem. Swarm of locusts attack crop across in Saudi Arabia. There are millions of locusts invade farm in Saudi Arabia and Yemen. And I'm sure it's not only in the Middle East, also in India, Australia, China, and other countries related. So problem segment is a locust population reach a certain density and eradicate beauty of environment. So you can see at the right side, so how when the swarm of locusts attack the city and the crop plantation in the Middle East. So, uh, so Moringa, aloe vera, and Cabra odolum is the have a waste here. Okay, nobody fully utilize the benefit of this plant. So I'm trying to integrate all the potential plant to become is a biopesticide uh, plus high frequency ultrasound, supersonic ultrasound. So and also because of the using of high dose for emitting locusts, and it's become worse at the area. It's not only million, now it's become billion and trillion at the specific area. You can see this is under remote sensing. Okay, so in 2018 and 2019 and 2020. So until now it's 2021. You can see the movement of the locusts. So you can see how the increasing of the population of locusts at the specific area. And for your info, it's not Saudi Arabia, but these locusts come from the Africa and with accompanied by the cyclone. So we can see later. This is a desert locust, okay, individual, and actually in this specific locust have come with the swarm or in the group, a huge group, and attack the specific area, especially in the crop plantation area. So you can see the swarm of this desert locust. So the objective of our study, for your info, actually this study is a collaboration between both countries, Malaysia and Kingdom of Saudi Arabia, and we want to see to investigate the potential of Moringa oleifera and Cabra odolum and seed extraction for locust seed pilot. Second, to study the effectiveness or extraction of moringa and Cabra odolum to increase eliminate of locust population. And third, to investigate the effectiveness by using integrated high frequency of supersonic to eliminate of locusts. So this is the movement of desert locust. So you can see this is at the right side, and desert locust situation update in 23rd April 2021. So how aggressive this insect when attack the specific area, especially in crop plantation. Okay, I don't want to go to further about what are the characteristic of the lockers. For your information, for your info, okay, how many lockers in one swarm is about 40 to 80 billion in each square kilometer. Okay, so and then flight speed is about 16 to 19 kilometer per hour, and average travel distance is about 5 to 150 kilometer per day. Okay, this speed 16 to 19 kilometer per hour is normal, but again when is with a accompanied by the cyclone is can go too fast, is more than 50 kilometer per hour. Okay, food consumed on daily is same at their weights, and how many days it occurs lay egg. Okay, it's about 95 to 158 per pound. Okay, locusts harm to human? No, not harm to human. But again, when it comes with a group, a huge group, it can attack your body, and you cannot do the breathe. Okay, good breathe, and then you will die again in the group locust. Okay, this is the summary of the locust characteristic. Okay, you can see how the locust destroy the agriculture within 15 to 30 minutes, not more than 1 hour. It's about 1 hour finished already. So, and it's harm to especially for the farmers, and the yield of production can decrease accordingly. So why I want to integrate the plant, the tree plant, okay, and the high frequency supersonic sensor, okay. So because each insect or uh, uh, have a specific frequency, and we can eliminate using the high frequency for eliminating the insect. So and then for for us, okay, human ear can perceive sound within a frequency range 20 hertz to 20 kilohertz, which is very limited. Okay, human ear is not sensitive to ultrasound. because the eardrum cannot vibrate as fast as ultrasound. Okay, so you can see uh, the range, okay, of penny locus is between 1 to 30 kilohertz. Okay, for locust 1 to 30 kilohertz. Grasshopper is 1 to 100 kilohertz. Okay, this is based on the literature study. Okay, so now our group from University Malaysia Pahang, okay, Faculty of Civil Engineering and Technology, University Malaysia Pahang, focus on the range 1 to 30 kilohertz. We want to find the specific, the right frequency for this locus. So when we found the right, the exact frequency, our hypothesis shows that the all the locus will settle down at the bottom, and what will happen, we can use this locus to others beneficial uses. Okay, this is about locus hearing system. Okay, locus have a highly integrated and miscellaneous hearing system that bears little resemblance to either the human ear or an electronic microphone. Okay, use of a nanostructure eardrum to discern between high and low frequency sound. Okay, locus eardrum membrane responded to incoming sound waves that were produced by a loudspeaker. Okay, for your info, okay, our team focus on a folded body (FB) and tympanal membrane (TM) in the abdomen of the locus. And then the membrane is kidney shaped and has 2 points on its inner surface where mechanoreceptor cells, neurons that respond to the mechanical stress, are attached in two different groups. Send the locus stays away from pulse ultrasound that simulate bad echolocation signal. The phonologic response was measured as ruddering by the abdomen, as I mentioned just now, and hind legs, resulting in a yaw tone directed away from the sound sources. Wing-beat frequency increased by 15% in response to ultrasonic stimulation. This behavior is considered to be analog to the bad avoidance behavior of flying crickets. The avoidance response was observed for carrier frequency higher than 10 kilohertz and for sound pressure level on average higher than 45 decibel sound pressure level. The avoidance response was suppressed when a 30 kilohertz normally effective tone was combined with a 5 kilohertz tone, which is ineffective alone. So this is a sensor that our team from University of Malaysia Pahang use for locus elimination. Okay, this is not my secret, but I have one friend is expert in the sensor, especially in the high frequency supersonic sensor. This is a diagram that we use in this research. These are the schematic diagram, okay, to show that when this sensor, okay, conducted at that specific area, okay, we can see the locus will settle down at the bottom. It's not die, but it's settled down bottom. So what's next? So we can use these locus to become other beneficial use, as I mentioned just now, because the locus itself have about 30 to 35% of protein, and it can become the protein sources for other beneficial uses like animal feeds and what call for the what called biofertilizer, compost fertilizer, and others raw material for protein sources. And it's very good instead of negative impact. But again, when we can control the locus population and the local attack, and we can change it from negative to positive view, which means it can come as a raw material for other beneficial use. This is a preparing of raw materials as repellent, as I I mentioned earlier that this is called Moringa oleifera, or in Malaysia, local name called drumstick tree or pokok kelo. And the other one is Cabra odalum, local name called pong pong. And others two natural plant neem tree is a Malaysian name pokok mambu, Azadicrata indica, and the other one also Anona muricata. But in this case, we have a certain stage, and this the first stage we're using drumstick tree or Moringa oleifera and pom pom tree or Cabra odalum. This is a preparation of the what called the potential what called plant, okay, raw material, especially for moringa leaf, uh, neem leaf, pong pong, and which mean pong pong fruit and leaf have a potential for eliminating locusts. Okay, this is grinding, it's become a small, okay, then the mechanism of using powder in a powder form is integrated with the high frequency supersonic is workable enough. Okay, this is that way. Okay, very simple. It's become my first stage before go to the pilot plant in the Middle East. So I'm using the small lab scale case to show that using the what called the high frequency supersonic have a potential for eliminating locusts. In this case, we because local has a this similar, it's not same but similar characteristic with the grasshopper. Okay, but the grasshopper in Malaysia is called belalang kunyit, is in the single activity. The locals have a in the swarm, in the group activity. So it shows in the lab scale, it shows that when we integrate the plant and the high frequency supersonic, it shows that we can eliminate the grasshopper accordingly. Desert local life cycle, I think I'm not go this further. All right, when we want to do the study, we must understand the behavior and the characteristic of the locus itself. Okay, so and then this okay, you can see at the schematic diagram below shows how these locus activities and swarm, band, group, adult hoppers activities can harm to the farm field, especially. And then is also have in the Pakistan, also Ethiopia, Somalia, and also in India. Okay. This is what called the different lockers in each country. In India, called Padanga Sunsinta. Okay, China called Kererik Kiangsu. Okay, and then Egypt, Anacrydium Aegyptium. Okay, Australia, Clotoicitis demifera, and desert or Middle East, Kingdom of Saudi Arabia is Schistocerca gregariana. So how they control and element process of desert locusts like this. You can see maintenance, hard maintenance, and then the spray with the chemical. So until now, no only organic or green technology solution apply in that specific area. So this is a common insecticide using to control the locusts in the Middle East. So insecticide organochlorine, organophosphate, carbamate, synthetic is class 2. When we deal with a class 2 of the pesticide is harmful. It's not only for human but also in animal itself. So we try to avoid to using. Now we want to good pest management GPM. So we try to eliminate, we try to reduce the using of the chemical for eliminating locusts at the specific area. So actually one of my product, if you know about last year, I already present about this product, have using the Deris elliptica extraction and have an active ingredient called retinol, have a potential for eliminating 10 insect in our in our environment. Okay, this product Pescila have patented already. I got the idea for eliminating locusts in Middle East based on this product, and but again, this product I'm using the retinol, the active ingredient from Deris Aptica, but again. For locust, because locust is aggressive, is a activity, is in a swarm activity, and it's not only million but also in billion and trillion. So we need to using the green technology solution, which means integrated within plant and juga device called high frequency supersonic. Okay, I go fast this, and then because I will, the Pesquila is already market and commercial. It's not only in Malaysia but also in 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 uh, in the Hojai. If you know this product Pesquila, I'm using this what called this solution to apply to the what called the agarwood tree in Hojai. Okay, it's about 1 acre in the pilot plant study, whereby that area is attacked by the white ant or termite. Okay, this product. Okay, why I want to highlight about the Pesquila product, because this product will has become a benchmark for my study regarding education plan for locust elimination in Middle East. So effective and easy to use, does not damage item, environmental friendly and safe and user friendly product from natural raw material, and no harmful to children, no harmful to environment, no harmful to the human. So this is a type of insect that we can eliminate by using the Derisaclitica plant. Okay, my company, okay, UMP Incorporation is one of the spin-off company for University Malaysia Pang. Already got international standard organization certificate, quality management system, environmental management system, and also ISO 45000 or last previous name called OSAS 18000, which means occupational safety and health management system. Why I get this certificate? We want to because we want to show that our company that produce and commercial green technology solution will penetrate market. It's not only in the local Malaysia but also in international level. And this is the achievement of the in the news. Okay, so many award. Okay, so okay, this why? Okay, I believe that using the moringa, aloe vera, and also the cabera or the lemon, because this based on my experience in using dairy cellulitica plant, because the what called active ingredient itself in each specific plant have a potential to eliminate locusts in that specific area. Okay, same like this. Okay, locust I show just now. Okay, this is in India. You can see many many what called activity or to make sure that the locust is can repel from that specific area. In Africa, okay, you can see spray, okay, with a chemical and so on. So, and then I'm already last month we using the Deris Antarctica. It shows that potential for eliminating insect in the Middle East, and also in the integrated of specific plant with high frequency supersonic shows that have a potential to to eliminate locusts in that area. So for your info, last month we already signed a memorandum of agreement with one big company in Jeddah, and Alhamdulillah they order about 3 container of Pesquila to make sure that this product, green technology product, can penetrate market in the Middle East. As I mentioned just now, okay, this is the beneficial users. As I mentioned, when the locus is settled down, we no need to what all to eliminate it simply like that. We can use it as other beneficial use for, and then because of the high content of protein, so we can become a raw material for protein sources. And then second, animal feed, goat, camel, sheep, horse, chicken, and many more. And third is compost organic fertilizer, biofertilizer, and also we can become a snack food. It's very, we don't know, and then maybe can become the powder, and then that powder or protein sources can become, for snack food and others. Okay, so this is name. Okay, I don't want to go too further because this is a potential of Azadirachta indica, or name, a local name in Malaysia called name, for eliminating workers. Okay, name food, name leaf, and name tree there. Okay, characteristic. Okay, so I don't want to go further. Okay, so okay, name all. Okay, type of soft body insect. Okay. We focus then, as I mentioned earlier just now, we focus about the hearing system of the locus, especially at the abdomen. Hearing locus is in the at the abdomen, in the abdomen. So we try to interrupt according to hearing system of the locus. And again, how about the powder from the green plant? So the powder green plant have a specific active ingredient, and then when the what called the locus attached by powder, and is will it's not die but will fainted, and then with the help of a frequency from the ultra supersonic, and it can work, the meaning that all the locusts will settle down in the bottom, and at that time we can collect it and process for other sources. Okay, this is using the name control. Okay, how name work as pesticide. Okay, have a certain mechanism. Okay, a certain mechanism to eliminate them. Okay, so many studies shows that neem have potential as become a biopesticide for insect repellent. Okay, supersonic ultrasound frequency using green technology solution for locust control. Okay, because these locusts have a specific, and then our study now to find out from the 1 to 30 kilo hertz frequency, we want to find out the right frequency, and to eliminate these locusts. And then many studies shows that ultrasound characteristic can affect animal, and can when can affect the pest, can affect a human, and so on. So okay, so in this case, the characteristic of our device is important aspect of ultrasound pest chassis is power output of the transducer, okay, direction and angle of sound, okay. The response depend on the number of pests in the population and the number of wave reaching in their vicinity. And some animal will ignore in the US initially, but later there's over. Okay, actually for your info, locust elimination study is already done. It's about 10 to 10 years in the Middle East. But again, now we want to study to show that the green technology solution can eliminate locusts, especially in our study location at the Middle East. Okay, locust hearing system as I mentioned just now. So, local eardrum, as I mentioned just now, is about the vibration. And then now one group of our team focus on this one eardrum of the locust uh, uh, physiology. Yes, ultrasound mechanism that can affect locus. Yes, sure. And then our study shows that uh, the study between 1 to 30 kilometer hertz, okay, shows that now we narrow down and then we find out that 10 kilohertz and for sound pressure level higher than 45 decibel specific pressure level can have a potential to eliminate locus. As a conclusion, okay, with a limited of time because the organizer gave me about 30 minutes, okay, to present about our study okay, from my research called locus, prevention locus. Okay, locus is the one called trademark of my product for this specific study. So as a conclusion, moringa and cabra are the pesticide concentration that will be effective to control locusts and can be used an alternative biopesticide in the future for locust elimination. And supersonic products from the device had a marginal effect in repelling locusts, and that's why the integrated of moringa oleifera, cabra odorum, seed pesticide, and ultrasound high frequency supersonic have a huge potential for elimination locusts, especially in the Middle East. That's all for my presentation today, and I open to the Q&A session. I pass to the Dr. Achuman. Thank you.
Thank you very much, Dr. Shokor. I invite any questions from our delegates if they are interested to ask any questions to Dr. Shokor.
Yeah, Dr. Abdul, I have to ask a query actually, not exactly question because it's not my area.
Yeah, yeah,
But you talked about Moringa and Cerbera. So
yeah, yeah.
the compound extracted from the Moringa and Cerbera were the part and parcel of your pest killer, or pest killer is something different.
Yes, the product called Pescilla is something different. That is from the extraction of Dadis elliptica. So Dadis elliptica, the active ingredient called retinol itself. So it's different. But again, as I mentioned just now, the Pescilla become a benchmark for our study in this integrated of Moringa and also Capera odorlum with high frequency supersonic. So different. Okay, but again, Pesquila already market in Malaysia since 2015. So already in the market is about 6 years.
No, in your presentation, you demonstrated many of the benchmarks. Pest killer is also one of them. But
Yeah,
my concern is what is the bioefficacy level of Moringa and Cerbera formulations?
yeah. Okay. Thank you very much for your question. But the bioefficacy for Moringa and Capra odorlum, we not studied yet because this project is already done since early, sorry, for the middle of last year, 2020, during the COVID. But again, we using not locust but also grasshopper called belalang kunyit in Malaysia for lab scale, a small lab scale. So for bioefficacy study, we not done for moringa and still in progress. Still in progress. Prof.
Okay, thank you.
Thank you. Okay, welcome. So any
Thank you
question
very
more?
much, Dr. Abdul. I don't see any questions coming up. Um, if any of our delegates wants to ask any question to Dr. Abdul, you're most welcome for the same. Um, okay, there's a question. From where does this locust come?
All right. Okay. From very good question. Okay. For my study, especially in the Middle East, I'm already done for what called study is about 1 week in June. So actually, then blockers not come from Middle East, but again, as I mentioned earlier, these locusts come from Africa. Okay, come from Africa, which is accompanied by the cyclone. Okay, as I mentioned, the characteristic of locusts is the movement is about 5 kilometer per hour to 15 kilometer per hour is in normal case. But again, it's with a cyclone, it can go further to 50 kilometer per hour, 200 kilometer per hour with the cyclone accompanying. Again, okay, this locust is very fast because of if you see that the study of the characteristics of locust is because of the raining season. Every month in Africa have a rain when rain, so the population of locusts become more and more. And when it become adult and cross the ocean to attack the crop plantation, especially in the kingdom of Saudi Arabia or the Middle East. Now our study, because now the team of sensor look into why the locusts can cross the ocean, the potential, the capability to cross the ocean to attack the crop plantation still under my study, and we do the what call we do the what call baseline study according to a report from US researcher in the Middle East. Yes.
Thank you, sir. Any more questions from our delegates to Dr. Shokor? I don't see any more questions coming in. So thank you, Dr. Shokor, for your great lecture
Thank you very
on
much. Okay, well, have. Thank you very much.
locust. Okay, thank you very much, sir.
Thank you very much for your team committee to invite us to share our study with all of you. Thank you very much to all. Thanks.
Thank you very much.
Thank you, sir. So now I would request our honorable chief guest for today and the keynote speaker, the former DG of UPCAR, Director General of Uttar Pradesh Council of Agriculture Research, and President of Indian Society of Genetics, Biotechnology Research and Development, Agra. Sir is now serving as the Principal Scientist at Division of Genetics at IARI, Pusa, New Delhi, ICA. Sir, over to you for your presentation. We warmly welcome you, sir.
Okay, thank you, Ansuman. How much time you are giving? And can you enlarge the screen? I mean to say, can you cover full screen? Or you are having some problem? I
Yes, sir.
dropped a message to you. Did you see my
Okay,
message?
sir, you can please. I didn't
Yeah.
saw your message. Sir, can you please email me your presentation?
Okay.
Once you email me your presentation, I'll share it from my side on screen.
Okay. I think I have sent you. Should I share or you will share, Ansuman? Hello, Ansuman.
Yes, sir.
You will share or I should share?
Hello.
Hello, I should share or you will share? Uh, hello, hello, hello. Hello. There is some problem. Hello, hello.
Sorry, sir. Sorry for the interruption. It was raining. It's raining here, so I just got disconnected due to some network issues. Uh,
Okay.
yes, sir. Sir, if I'll just check my mail. Yes, sir, I got your presentation. I'll share it from my side, and you can just continue with your presentation. Just give me a minute. I'll
Yeah,
start sharing the screen.
no issue.
Yes, right. So we are ready with your presentation. I'm starting to share the screen, and with this, here we go. Um, share screen, share screen, share screen, allow. Yes, sir. Are you able to see the screen now? Your presentation?
Yeah, yeah, I'm able to see the scheme.
Yeah.
Good afternoon, Dr. P.K. Upadhyay, Secretary of the Society, Dr. Ulrich, Dr. Abdul, and many dignitaries in the studio, and dear audience across the country as well as globe. Today, first of all, I extend my thanks to Dr. Upadhyay because he compelled me to deliver the lecture due to some personal health reasons. For last few months, 5 6 months, I was on rest. So, anyways, he forced me to deliver. So, my deliverance is diligently potential resources for micro and macro nutritional genetic gains in food legumes. Next slide. As far as food legume is concerned, everyone is aware that food legumes global coverage is at 9 million hectares with a total production of 88 million tons, but having very low productivity per hectare. It's only 992 kg. The cultivation of food is advantageous for succeeding crops because it fixes the atmospheric nitrogen ranging from 20 to 40 kg, and also it's very rich in protein. Besides protein, it had also got many of the antioxidants and beneficial nutrients. It's grown across the globe. Of course, it has a group of legumes, more than 35 legumes uh, across the globe. Next slide, please. So we can see how much variability as a group of the pulses. Around 35 major or minor pulse graphs are grown in different parts of the globe. Next slide, please. As far as India is concerned, India grows 78 major pulse graphs with a share of about 34% of the global area, and with a share of 24, more than 24% of the total production to the globe. The irrespective of being the traditional grower of the food legumes, its area as well as production and productivity always varies. But as for FAO 2019, the area coverage is 30.82 million hectares with a total production of around 22 million tons, and productivity once again very low, that is 698 kg per hectare. The imported food legumes growing in different parts of the country are leading countries are Madhya Pradesh, Maharashtra, Uttar Pradesh, Rajasthan, Andhra Pradesh, Karnataka, Gujarat, Chhattisgarh, and Bihar. Particularly confined to gram, arhar, mung bean, urd bean, lentil, peas, and harsh grams, including moth. And these areas of pulse cultivations actually taking a shift because earlier, a few decades back, North Indian states were the major growers, but now its area is shrinking in the north states of the country. And but at satisfactory point is that it's expanding or taking more coverage in the southern parts of the country. The desirable nutritional attributes of food legumes are several. It provides us high protein, high dietary fiber, low fat, low glycemic index, gluten, low gluten, and also very good amounts of antioxidants and good quantity of the energy. Besides, many of the micronutrients, and there are some of the pulses which are very good as a source of the anti-diabetics as well as anti-cancers. Besides, these are cholesterol free. Next slide, please. Potentials of the food legumes as nutrition are several because they are the good source of the nutritions. They can aid to the value-added products in several forms. Besides, there are several therapeutic values and bioactive compounds as well as functional properties. Next, food legumes as a source of protein that is well established. Many diets around the world rely on food legumes as a source of protein. The amount of protein in beans, lentils, chickpea, because why I'm talking about beans, dry beans, and chickpea, these and peas. These are the major crops of the global contribution. So these contribute to more, 2-3 times of the level of the protein as well as other nutrients uh, in comparison to the cereal grains like wheat, rice, quinoa and oats, barley, and corn. we can get enough amount of proteins just out of half cup of lentils. That goes equivalent to 1 cup of quinoa or 2 cups of the rice or corn. Compared to animal and many other plant-based sources of protein, food legumes are more affordable and sustainable source of proteins in the longer days. Next slide. Next slide, Ansuman. Food legumes, no, back, please, back. Let me say, whenever I say, only then you roll out for the next slide. Food legumes are also excellent source of fibers as well as complex carbohydrates. Besides, they give enough soluble as well as insoluble fibers for the daily intakes. Food legumes also contain resistant starch, a type of carbohydrate that behaves like fiber in the body, as has shown health benefits such as reduced circulating cholesterol and blood sugar levels, besides improved gut health. Next slide, please. We can see how much variability as far as nutritional components are concerned. I am presenting you the data of the USDA, United States Department of Agriculture 28th release. We can see there are number of the nutrients ranging covering protein, fat, carbohydrate, dietary fiber, minerals, vitamins, lipids, etc. Particularly of the major crops like soya bean, peas, lentils, lupins, chickpea, white bean, and black bean. So we can see how much potentials for the micro and micronutrients are hidden, yeah, as far as pulses are concerned, or food legumes are concerned. We call also food legumes or pulses. Next slide, please. Here I am concerned with the genetic gains. I take you back to the deliverance of Dr. Ulrich as well as Dr. Abdul. They talked about the limitations of the nature. The problem is that if we, by the as per the FAO report, by 2050, we are going to have nutritional crunch, not food crunch. But now we have started talking about the nutritional crunch. So by 2050, if we have to really live the life of the United States, we require at least 5 planets, which is not possible. We cannot create the planets. Besides, we are losing our natural resources. I mean to say sunlight, air, and water quality. Dr. Ullis also talked about the saline, alkaline, sodic, and all those lands. Besides acidic lands, so there is a challenge before us. And besides, there are several infestations of the insect pest, particularly about the locust. Now, Dr. Abdul was talking about the locusts. Locusts are not confined only to the Malaysia or Arabian countries. Rather, in the past years, we have observed heavy infestations of the locust even in our Indian scenario also. They reach to us also. So certainly there is a challenge. So in the light of the shrinking resources, I mean to say air, land, water, and other natural resources, how to cater the requirements of the humanity as a whole? Because we cannot design new living systems suddenly. Of course, we are trying to create the variability, or we are adopting the transgenic approach even for the animals as well as plants and everywhere. But the limitations are there. So now, as a bidder, we have started talking about the genetic gains. How the genetic gains can be harnessed? How we can enhance the genetic potential of already available biodiversity in the nature? So genetic gain is the term used to measure the genetic improvement or breeding progress and can be defined as the amount of increase in the performance that is achieved annually through artificial selection and is measured by difference between a selected population and their offsprings in the near future. Next slide, please. Whenever we are talking about the genetic gain, there are several factors which actually influence the genetic gains. Like uh, inbreeding, seed increase, multi-location trials, multi-year testings, and we have to predict the phenotypic performance also. Besides, cheaper to genotype larger populations for the same selection intensity, making selections even during the off-target years, and trying to have genetic gain over time, and combining favorable rare alleles at one place across the different years. So we can say we can see this formula genetic gain over time that is represented by RT, and selection intensity that is represented by I. Then come the selection accuracy. What is the accuracy of the selection, particularly during the off years or off target years? And genetic variance, available genetic variance in the nature across the year. So these are the certain factors which are really going to influence genetic gains, and we have to be very cautious. I will be talking more in the later slides. Next slide, please. So factors, these are the genetic variants, these subcomponents. Once again, these can be compartmentalized in different parts, like germ plasm should be exploited properly. Molecular markers, which we are also consider as a genetic resources, they should also be exploited in combination with the genetic variants. Genetic parameters used for the estimation of the genetic variants and creation of the genetic variants. Then heritability, population used for estimation of heritability. Whenever we talk of the heritability, actually the high heritability is more desirable, and for estimating accurate and high heritability, we have to go for the multi-location trials for having the phenotypic observations for getting exact variance of the phenotypes. Estimation of the environmental variance, applying proper statistical tools, which I was talking about during the oration of Dr. Ulich also, because unless until our statistical methods are accurate and they are going to make our finding robust, none of our findings are going to be rewarding in the longer day. Then comes the selection intensity, how rigorous selections we are going to make, and what is the cycle of the time. So in nutshell, the duration of the cycles, life cycles are here. We are talking the life cycles in the duration of the years. Selection intensity, I mean to say larger should be the population. Then not only larger population, rather multi-location trials to minimize the environmental errors, and also exploiting the larger population from the germplasm. Uh, so these are the certain factors which contribute very significantly for the genetic gains. Next slide, please. So if we summarize to that, we can see the major challenges, the genotypes which are available with us, they are having low genetic potentials. We have to increase the genetic potentials, and the low realized yield, changing climatic situation. We expect that every time what is the rate of our climate change, and besides biotic as well as abiotic stresses in different forms, and which is easily manageable, that is poor seed replacement, post-harvest losses. We all know that the post-harvest losses contribute very to significant. In case of vegetables, it contributes around 30 to 35% of the post-harvest losses. Even in case of the cereals and others, minimum it contributes for the 7% at least losses. And poor transfer of technologies. We have several technologies, but we are not able to ensure their awareness among the farmers and get them acquainted for adopting those proper technologies. Next slide, please. Challenges for genetic gains in food legumes are like negative correlations between yield and protein. It's not confined only to high yield and high protein. Uh, Rather, there are certain many negative correlations in different traits. Whenever there is high yield, then protein content is low. When there is protein content is high, then yield is low. We have to break that negative correlation. Besides, there is a negative correlation between protein and sulfur containing amino acids. And moreover, Moreover, nutritional traits are being governed by the polygenic polygenes. And the most important is we are lacking the systematic nutritional analysis. Uh, we are so what is the problem? You just Ansuman, do you have any problem? I think yeah. Okay. So lack of systematic nutritional analysis. The unfortunate situation is with the new advent of the new technology, biotechnology, almost all biochemists and physiologists, they have turned to be the biotechnologists. So we have to focus for the phenotyping for the nutritional traits. Unless we have accurate phenotyping for the nutritional traits, we cannot enhance or harness the genetic gains. Besides, we have also to develop the proper field screening techniques for nutritional traits. So these are certain challenges. Next slide, please. Prospects for enhancing genetic gains in terms of negative correlations have been recorded in several of the traits, as we already discussed. Even then, there are certain exceptions, and these negative correlations can be broken, and positive correlations can be developed. It by adopting the approach of the mutagenesis or redesigning the genetic recombinations uh, by different approaches. Next slide, please. Once again, we can see that food legumes are deficient in the sulfur-containing amino acids, particularly tryptophan, but they are very rich in the lysine, which is deficient in the cereal. So we can increase these amino acids through gene integration, through selections, wide hybridization, and other approaches. And biological values of the food legumes vary at length 30 to 78%. These biological values can be enhanced, particularly in terms of the low methionine content. There is a scope for increasing the biological value of the pulses or food legume as a whole. There are many reports that biological value of grain chickpea varies 52 to 78%. In case of urd bean, it varies 60 to 64%. In case of pigeon pea, it varies 46 to 74%. Lentil 32 to 58%, Moonbeam 39 to 66%, Pea 48 to 49%. So at least there is a satisfactory point that the one of the major food legume, that is chickpea, it has very good biological value variation. So these crops can be exploited for enhancing the genetic gains. Next slide. we can see concurrent choice of both yield as well as protein or other desirable nutritional components, whether we are talking in terms of the antioxidants or macro or micronutrients. Certainly negative correlations exist, but these variations can be exploited and they can coexist. There are certain reports, particularly in case of maize, OPEC2 gene have been observed and protein contents has been enhanced at the large level in case of maize. Such type of strategies can be adopted even in case of the food legumes, and by utilizing the wide hybridization or wild genotypes of the food legumes. So with such a range of variability, we can have interfamily selection for yield, nutritional traits, protein, etc. So and so, and ill effects can be mitigated. Next slide, please. Accordingly, in the food legumes, the negative correlation between protein and protein content, methionine and cysteine can be modified, as it has already been demonstrated in case of cereals. Apart to this, as in cereals, several mutants with higher protein content and amino acid profiles have also been identified in case of food legumes also. Appropriate genetic manipulations of these nutrients are required to bring about an appreciable improvements in both protein as well as other traits of interest in quantity as well as quantity both. Next slide, please. So we can, besides these genetic improvements, we can have also we have to refine our field screening techniques for the nutritional traits. We can have several of the field screening strategies, like many procedures for a chemical analysis of the protein and amino acids have been developed, like IR reflectance. Besides peptide analysis by the mildly mass spectrometry, amino acid analysis, protein structure, HPLC with a detector IR, amino acid analysis system, and FTIR analysis, sugars and oligosaccharide through bioscan. Besides mineral analysis, like with the help of double AS or inductively coupled plasma mass spectrometry. Besides improved field techniques, and even in the days ahead, there are new technologies are coming. So these field techniques are also being improved, and these can be clubbed with the plant breeding activity. Next slide, please. Sufficient variation exists as far as various components of food legumes are concerned, and they have satisfactory variability ranging from 1 to 40% for the protein content and oil content, besides carbohydrate ranging 2 to 60% and protein ranging 15 to 50% of their dry weight. Besides manipulation of proportion of these components are also required for different combinations of major components, particularly associated with the complex biochemical machinery, which are little known and they are required to be done. Next slide, please. So we have also tried to focus, for example, we tried a set of genotypes for the protein content, and we realized that there are 3 genotypes which can be utilized as a genetic donor. These are BG1092, ICC11378, and JG74. So two genotypes, one genotype belongs to our own material BG1092 at IRI, and ICC11378 is a from global germ plant of the chickpea, besides JG74. These are the 3 genotypes which can be utilized. With the highest protein content observed in case of BG1092, with the lowest one in case of Pusa 256. Once again, lowest for the IRI genotype. So the next slide, please. We have conducted another study on the nutritional aspects, and we observed antioxidant activity, nutraceutical profiles, and health relevant functionality of 9 genotypes. And we came to the conclusion that there are one of the genotype Pusa 1108, which is our own variety, released in 2006, that performed well for the antioxidant like DPP, HS, and TRAP, and they can be utilized in the breeding programs as a donor. Next slide, please. Besides, we conducted one another experiments also on the same set of the genotypes, and we identified that there was a good variation for protein, as well as, of course, here genotypes were limited. So the good content of the protein as well as antioxidants, and we concluded that amongst the Desi genotype K850, one of the variety developed by Chandrasekhar Azad University of Agriculture and Technology long back, and our own variety Pusa 1108, that's a Kabuli genotype, and Pusa 1088, once again Kabuli genotype, and Pusa 1053, once again Kabuli genotype. They can be used as a good source for the nutritional as well as anti-nutritional profiles in case of chickpea breeding program. Next slide, please. We conducted another experiment also for estimation of the nitrate reductase, and we came to the conclusion that highest nitrate reductase were found in case of JG11, with the lowest one in case of Pusa 372. And we also utilized a set of molecular markers, and finally we came to the conclusion that JG11 as well as F5 198 can be exploited in our breeding programs in the days ahead if we have to focus on the nitrate reductase enzyme. Next slide, please. Besides, there are certain problems whenever we talk of the food legumes. Here I am taking only chickpea as a study case because my area is confined to chickpea only. So that's why I'm taking chickpeas as a case study. So in case of pulses, we find many of the anti-nutritional components also, particularly oligosaccharides in different forms like raffinose, barbachose, and istachio. So we conducted an experiment. This was the experiment done by one of my PhD student, and we focused for the raffinose. We came to the conclusion that raffinose, there is one genotype of the global germplasm, that is ICC 1083, which can be utilized as one of the donor because it contains very low quantity of raffinose. Next slide, please. We also try to utilize the chickpea husk in case of other byproducts. So this was one of the trial. In case of biscuit making, we used this chickpea husk, and we came to the conclusion that antioxidants performance was very good in case of the biscuits when they were compared with the inclusion of the chickpea husk. Next slide, please. Besides, we also try to see the bioefficacy, I mean to say biological value in terms of the antioxidants as well as functional properties for the new developed varieties by us. And we came to the conclusion that there are certain genotypes which can be utilized in the breeding programs, besides not only in the breeding programs but even in the government sponsored food programs also, for giving better nutritional supplements. So here once again, Pusa 1108 gave very good performance, and K 805 sorry Kanpur 850 they gave very good performance, and they can be utilized in the breeding programs as a donor. Next slide, please. Once again, we conducted for the digestibility of the different nutrients, and we concluded that when chickpea products were uh, subjected to autoclave, their digestibility and protein bioavailability was better than other mode of experiments like thermal subjectation or growing in the micro cooking, boiling, roasting, hydrothermal, etc. Next slide, please. Once again, we made another other experiment for the proteomic availability, sorry, protein availability, bioavailability of the protein in case of the chickpea. Uh, that too on the buried germination stages. And we came to the conclusion that three genotypes which were very good in case of protein, high protein content, like BG1092, IRI genotype, ICC 11378, and JG74. They also gave very good biological digestible values at the 3 days stage as far as human consumption is concerned. So once again, it's established that these genotypes can be utilized if we use germinated seeds. Certainly, our human food consumption is going to be efficient one. Next slide, please. This is one of the latest publication. Yeah. Besides this, I have already discussed in detail because it's too late. I think many of the audience and including me is also hungry. Next slide, please. So very recently we published this publication. We tried to assess the functional and nutritional properties of the chickpea genotypes, and we came to the conclusion that protein content in general were high in case of Pusa 1108 and in case of Kabuli. And whenever these were used as a isolated as a protein isolate and used as a byproduct in case of our human diet, the efficacy of these products increased manifold. So this chickpea genotype, particularly 1108 and Kanpur 850, can be utilized even in the case of protein isolate preparations. Next slide. We can see these are the only two genotypes and how much variability we have found. We find that almost all essential amino acids are available in case of chickpea. Of course, as far as total availability of the chickpea is concerned, desi type chickpea was giving better output than Kabuli chickpea, but digestibility of the Kabuli chickpea was better than desi chickpea. And we have to refine the strategies because one of the report given by the Wang et al. Reports very high content of the nutrients like in case of desi as well as Kabuli, but we could not found those such type of high quantity of the nutrients. So I think more efficient protocols and procedures have to be adopted, so that can be automated and faster experiments can be conducted as far as nutritional phenotypic is concerned. Next slide, please. Uh, Very recently, we also conducted an experiment for the estimation of the superoxide SOD enzyme, which is very important for the stress uh, resistant genotypes. So we found that PUSA 200, 366 and one of the genotype ICSET 3668, these gave the maximum variation. And our study, even through genotyping utilizing the molecular markers, came to the conclusion that ICSAT 3668 can be utilized one of the donor for the SOD enzyme. Next slide, please. Besides, next slide. This is one the dendrogram or the genotyping based utilizing molecular markers. So we came to the next slide. We recommend TA80. Next slide. We recommend TA80, which is highly polymorphic and shows association for the SOD content. Besides, these are the certain genotypes which can be utilized as a donor, like ICSET 3668, CSG 9505, SBT 377, and BGD 72. These are the genotypes which can be utilized as a donor for the SOD content in the future breeding programs. Next slide, please. Very recently, we have estimated certain genotypes for 12 traits. And we observed that there is a good variability available in among these genotypes. And even our standard error, because these experiments were conducted at the multi-location trials, and besides we replicated trials, and we find that enough variability is available, which is supposed to be stable. So we also ran these populations for the next slide, for the molecular analysis also. And a comparison of the molecular as well as phenotypic dendrograms were made, and we conclude. Next slide. We conclude that out of the 73 utilized markers, 10 markers revealed good polymorphism as far as nutritional traits are concerned. And out of these 10 STMS markers, we had designed 27 new markers for the first time reporting. And these are SBP 55, SBP 95, SBP 96, SBP 146, SBP 213, and SBP 217. And five previously reported markers by Dr. Vasle and it's all Dr. Rajiv Vasle from ICSET, and two genotypes, especially in the Desi group, PUSA 1103 and Kanpur 850, and in Kabli group, PUSA 1108 and PUSA 1053. These are the genetic resources which can be utilized as a molecular markers as well as donor genotypes for the macro and micronutrients analysis and further studies. Next slide, please. As far as heritability is concerned, of course, once again, heritability is one of the major factors which I have narrated earlier in one of the slides. That if heritability is high, then genetic gain is also high. In case of pulses, the genetic gain is not very good. It's around 0.9%. So this, in general, heritability is supposed to be increased. For that, multi-location trials, accurate selection process over the years, uh, intensive selections are supposed to be made. Next slide, please. How we can enhance the genetic gains in case of the food legumes, like bigger programs, particularly mechanization, automation, digitization, with the higher selection intensity. One of the requirement. Second requirement is the enough genetic variability should be available to be utilized as a donors, particularly from the elite but exotic materials, and more accurate selection. I mean to say, intense selections and accurate selection for getting higher heritability, especially utilizing the marker-assisted selection, better experimental designs, higher quality phenotyping, and robust statistical tools. Faster breeding cycles, because in a shorter duration, if we have to deliver the products and in order to cater the needs, we have to make proper genomic predictions, and we have to design certain programs. So we have to hasten the breeding cycles. It can be done by the speed breeding or the off-season cultivations, whatever may be, those have to be adopted. Besides management that is empowered and accountable for product delivery, once again, multi-directional activity that has to be managed from agronomic practices to insect pest control, I mean to say integrated pest management, etc., and coordination among various stakeholders from biochemists, insecticides, entomologist, pathologist, breeder, geneticist, etc., so and so, and overall well-trained staff who understand how products are produced. So in order to that, all stakeholders, including plant breeders, geneticists, tractor driver, entomologist, pathologist, each and every stakeholder involved in the agriculture as a whole, they are supposed to be trained. Next slide, please. We can see, if we talk particularly in terms of the genetics and plant breeding, gene bank. Gene bank basically conjures, characterizes, and distributes germplasm researches on germplasm to identify trait-specific lines. It needs to be intensified. Pre-breeding program to characterize germplasm and develop suitable material, and identify novel genetic populations. Pre-breeding has to be strengthened. Genomics and trait discovery, like genomic resources, comparative and functional genomics approaches, new alleles discovery and markers, all those trait of interest have to be intensified. And we have to go for the forward bidding with the help of the markers, clubbing with the backward backward bidding also. Because whenever we have to apply for the back cross bidding, we have to adopt the approach. Besides, hardcore cell molecular biology and genetic engineering also needs to be harnessed, like induced variants for the bidding programs and transgenic developments. And overall seed system has to be optimized, and it needs strengthening for the delivery of the seed system, including uh, transfer of technologies at the farmers' fields. Next slide. So we can say, now our breeding strategies have to focus around uh, climate resilient, nutritional, and market oriented traits, large-scale germplasm sequencing and value addition, novel trait breeding lines, and compressing pre-breeding time. Candidate gene, QTLs, and diagnostic markers are must-have traits, and novel breeding approaches including tested, validated, and applied biotechnology and genomic approaches, double haploid and speed breeding methods, strengthening and modernization of the seed systems, new sequencing and genotyping platform, resources mobilization, enhanced capacity building and communication. Next slide, please. So we can see, by and large, we have discussed, but we just giving one of the chart presented by by you et al. 2017. We can say what are the parameters for the potential increasing, and what are the gaps which are required to be closed, like breeding pest resistant, soil stresses, water stresses, herbicide resistant, climate change, resource use efficiency. These have to be addressed by the bidders. Besides crop and field management, weed control, pest control, soil fertility, water management, mechanization, precision agriculture, and also socio-economic policy like cost, credit, tradition, knowledge, input, instructions, and potential increasing. We have to unlock and create genetic variation, improving heritability, like in we have to environmental envirotyping, error control, genotype into environment interaction, economic practices, heightening selection intensity, like sample size that should be the larger, the throw output, mechanization, automated, then precision and cost. Besides shortening breeding cycles by involving mass, double haplast, integrated, and speed breeding. So we can say that what is the situation? Situation is that actual, if we see what is the experimental station yield, we can see, or rather, let us come to this theoretical. How we imagine the theoretical potential says that right now we have overall 17.1 million ton per hectare potential, but on the experimental station trials, we are able to get or meet out only up to this level. I mean to say, here only, and once again there is a gap with whom actual farm yields. The farmers feel the total production per hectare is 5 ton. So what we see, there is a this gap, 17 minus 5. Accordingly, once again, potential farm yield. Here also there is a gap. So there is a huge gap between actual farm realization, potential farm realization, experimental reports, and theoretical potential. So we have to minimize these gaps. Next slide. So in brief, we can say, as a builder, if we harness the different approaches of the crop improvement, then only we can develop some new genotypes in a shorter duration. There are by and large 4 approaches. Like one is the wild crossing. I mean to say, one is utilizing the wild genotypes, making crosses with the elite lines, and we can see whether we are going by the uh, F1 back crosses or so, and or with the selfing. Then in case of wide crossing, adopting conventional breeding techniques, minimum 10 years, 7 to 10 years duration is required. If we apply molecular markers with the help for these things, these 6-8 generations we can get in a very short duration, and we can get these new genotypes in a shorter duration, like 3 to 6 years. But for the conventional breeding approaches, once again 7 to 10 years. And in case of genetic transformation, coupled with the speed breeding and molecular markers and other latest tools, we can have new genotypes released in the 5 6 years. So there is a, I mean to say, integrated approach is required. And if we integrate all these things together, Even we can get the new genotypes. I mean to say, transgenings are good potent genotypes even in a short duration of 5 6 years. Next genotype. Next slide, please. There are certain new tools like epigenetics, speed beating, editing tools, association mapping. Still, these tools are to be exploited. Now we can have a behavioral study of the nutritional traits and genetic behaviors, and these can be work as a mediator to enhance the genetic gain, even in case of the food legumes for the nutritional traits. Speed breeding, it can enhance the genetic gain by shortening the breeding cycles. If we adopt the breeding, the annual crops, which we are raising only one crop in a season, here we can have 3 4 crops by standardizing the crop duration age, by managing proper light and humidity, uh, etc., and temperature. Besides human association mapping, these association mapping can be harnessed for wide variability already available in the nature, and in case of certain materials where seed mating or mating is not possible, or the larger time is required for the different type of mapping populations. Even our genome map was also mapped only utilizing this association mapping. So this association mapping can be equally applied in case of the food legumes also. Food legumes also. So we can use these mechanisms to mediate or improve for these various nutrients. Next slide, please. So we can say these are the different tools which are required to be integrated at one platform. In the integrated platform, the Dr. Yunwei et al. They have presented in a very nice dramatic way. So once again, not many things, but only in integration of the mass marker assisted breeding, genomic selection, and high intensity selection, germplasm evaluation, new inbreds, seed or leaf DNA based marker selections, and field testing, etc. If we combine all these at one platform, certainly we can uh, minimize the duration required for development of any variety. Next slide, please. And genetic gain, as far as in case of pulses are concerned, we can say that right now we have limited genetic gain in case of food legumes. It's around 0.9, and in case of chickpea, it's around 1%. It can be enhanced even with the available genetic variability available in the nature itself. Only the thing that we have to make systematic and concerted effort for harnessing these potentials and to increase the genetic gains. Next slide, please. Next slide, please. I'm not going to, once again, that is only, we can increase this 1% to height by utilizing these potentials. Next one. So conclusion is that genetic gain can be increased by integrating all these approaches. Biofortification can be recognized one of the viable tool. Special techniques like in vitro cultures, chromosome-induced mutants, etc., can be harnessed, including mutagenesis, and several attempts for the amino acid profiling, including the gene editing, not only for the gene, rather editing for the amino acids or even for the nucleotide itself. These can be are the future tools which can be very potent. And if these tools are integrated and one platform, certainly we can increase the genetic potential as a whole. Thank you. Next slide, please. So these are our contributors. Many newcomers are left. Uh, so with all these, thank you everyone. And if there is any query and curiosity, you people are most welcome, please.
Thank you very much, sir, for your presentation. It was good to see you. I'm seeing many messages coming in the chat box regarding the feedback form. It is already shared in the brochure that the feedback form will be shared within a week and your certificate will be provided to you. Please do not share messages again and again. It will automatically be shared on your mail addresses. Thank you. Next in the queue, we have Dr. Shweta Thakur with us, who is the assistant professor at Department of Plant Biosciences and Department of Biosciences. And we'll continue with the talk. But one question is coming for you, sir. Professor Rajan Kumar, sir, one question is coming for you from
Yeah.
Dr. Pooja Singh. She is asking, Honorable Sir, please explain what is meant by transgenic male insect resistance.
Transgenic.
Male insect resistance.
Okay, transgenic male insect resistance. Uh, Actually, whenever we talk of the insect resistance, so there are certain genotypes, particularly those genotypes are uh, behaving as a resistant with the males only. Why males only? You have to take your focus on the X and Y. So in case of male insect resistant genotype, that is in the heterozygous condition. But in case of females, that is in case of the homozygous XX. That's why, and we know that whenever X is concerned, they are supposed to be in different behavior. So if homozygous X is lacking, only then you get the resistance for that. And for such genotypes, if we are having the resistance, then we can say these are the transgenics which are applicable only for the male resistance. In case otherwise, your population will finish. I think it's clear. Dr. Pooja,
Yes, sir. Yes, sir. It's clear. It's clear, sir.
any more question?
She's yes, sir. One question is coming. Honorable sir, please guide me where in India a genetics and plant breeding student can do training program for seed production.
As far as seed production is concerned, now there are a huge number of organizations. Almost all agriculture universities are involved in the seed production programs. Time to time, they conduct training programs also. But as a nodal center, we have one institute, Indian Institute of Seed Research, that is located in the Mau district of Uttar Pradesh. So, if students or any farmer, anybody is concerned, they can contact that institute because that institute is the nodal agency for the country. Besides IARI, we have C technology department. They are also involved in such training programs. Even Directorate of Government of India, Directorate of Agriculture is also involved in the such training programs. All state governments are also conducting such type of training programs. But directly, as far as the students are concerned, or farmers are concerned, They can contact respective deputy director agriculture in their area of zone. They can update them where there is a timely organization of such training programs. Otherwise, they can contact these two institutes, at least IRI as well as uh, Institute of Indian Seed Research.
Yes, sir. Thank you, sir. I guess there are no more further questions. Okay, no more further questions, sir. Thank you very much. And I am extremely thankful to Professor Rajendra Kumar, sir, as he was not well, but still he took out time for us and gave us such a great lecture today. And it was so great of you, sir. Thank you, sir. Thank you
Thank
very
you.
much.
Everyone, thank you very much. I have to take my medicine, so Sweta, I'm sorry, I'm leaving.
No issues,
Okay,
sir.
thank
Thank
you.
you, sir.
No, no, sir.
Shweta ma'am, can you please unmute your mic? Your mic is mute.
Yes, can you
Yes.
hear
Yeah, now it's
now?
good. Now it's good. Yes, ma'am.
Okay, okay.
Now we can
So
hear.
should I start?
Yes, ma'am. Please. Yes, I would like
Okay.
to give a short brief about Dr. Shweta Thakur, ma'am. Ma'am is the assistant professor at Department of Biosciences at Himachal Pradesh University, Shimla, and ma'am will be taking further with the lecture. Over to you, ma'am. Thank you.
Okay, thank you, Anshuman, and thank you, Indian Society of Genetics, Biotechnology Research and Development, for inviting me to deliver a lecture. And good afternoon to all esteemed speakers and audience. So my today's topic is, let me just, uh, application and advantages of genetic engineering technology for crop plant abiotic stress. So can you see it? So is it visible to all? Hello?
Yes, ma'am. Your slide is visible. Application and advantages of
Yes,
genetic
yes, yes.
engineering technology for crop plants and biotic
Cropland
stress
abiotic.
tolerance.
Yes, thank you,
Yes, ma'am.
Anshuman. So, as we all know, climate change and pollution are the major problems in today's world. So, everything is becoming polluted nowadays. And because of this, our crop plants are exposed more frequently to the episodes of abiotic stress, such as drought, salinity, elevated temperature, submergence, and nutrient deficiencies, etc. Sorry, wait for a minute. I think so. Sorry for the interruption. So, I'm continuing now. So, the due to the pollution and changing climate, the our crop plants are now more susceptible to being exposed to abiotic stresses. So abiotic stresses include major of them are drought stress, heavy metal stress, salinity, elevated temperature, submergence, and nutrient deficiencies, etc. So the abiotic stress cause severe damage to our crop production, our yield, and plant growth and development, and they can persistently limit choice of crops and agriculture production over large areas. And extreme events can lead to total crop failures also. As we hear lots of news like crop failure due to drought, crop failure due to flood, etc. So these abiotic stresses are adversely affecting not only the crop yield but also the livelihoods of individual farmers and their families, as well as national economies and food security also. So, like according to an estimate, due to the declining water availability by 2025, 30% crop production will be at risk of drought stress. And according to another World Bank report, the climate change will reduce the crop yield by 20% or more by year 2050. So, on the one hand, we need more crop and more food for our growing population worldwide. On the second hand, our crop production is declining due to the climate change and due to the effect of these abiotic stresses. So, therefore, the abiotic stress management is one of the major challenges in the field of today's agriculture research. So in this slide, you can see the major environmental factors and their effect on plant growth. Although the main effect on plant growth and development and crop yield of one or different abiotic stresses are almost same, but a few effects are associated with a particular abiotic stress. So I try to give them in a table like high temperature cause membrane and protein destabilization, photosynthetic and respiratory inhibition, ROS production and cell death. Chilling also cause membrane, it has a major effect on membrane destabilization and membrane dysfunction, which causes a plant cell to die because there are so many enzymes and proteins associated with membrane only. If a membrane of the cell destabilizes or it starts dysfunctioning, the cell cannot function properly and ultimately it leads to cell death. And next is flooding. It causes hypoxia, anoxia, reduced respiration, fermentative metabolism, inadequate ATP production, ROS production, and stomatal closure, etc. Freezing also causes water potential reduction, cell hydration, and symplastic ice crystal formation. And another major threat to our agriculture nowadays is trace element toxicity. We can call them trace element toxicity or heavy metal toxicity. So they are also very, dangerous pollutants, which are increasing day by day due to human activities only. And they have capacity to disturb co-fact binding to protein and DNA, ROS production, disruption of metabolism, and they mimic other essential metal ions. Like heavy metals are all, not all are toxic. There are some essential also. They need for normal plant growth and development. They are cofactor to various different enzymes in proteins, but the toxic one like nickel, uh, cadmium, they replace the essential ones, and thus if they replace in the essential one in enzyme, then the enzyme will no more be active or it will not be able to perform its function. And mineral nutrient deficiencies, because plant start to uptake the non-essential ions in place of essential ions, so it cause the nutrient deficiencies in plants also, which also hampers their growth. And next is ozone and ultraviolet light. It generate reactive oxygen species that cause lesion and induced programmed cell death. And they in the last slide I was talking about the individual abiotic stresses. As we work in our laboratories also, we focus on single abiotic stress, but in the field condition the conditions are entirely different. In field condition it's very rare that only one stress is present. In field condition the plants face multiple stresses at a time, because if there is heat stress, then there will automatically if there is drought stress, then there will automatically be water deficiency and heat stress also. So if there is freezing stress, there will be chilling stress. So they are all interconnected in field conditions. And the combination of abiotic stresses induce unique signaling and metabolic pathways as compared to the individual stress. So this is also a big loophole in our today's research of abiotic stress tolerance in plants, because we in lab conditions we focus only one abiotic stress, but the conditions are entirely different in field level. So we should start uh, studying the effect of multiple stresses, and we should focus on developing the varieties which can withstand the effect of multiple stresses. So plants also have their defense mechanism against stresses. So plants employ mainly three strategies to cope with these stresses. First one is avoidance, like they prevent the exposure to stress. Second is tolerance. They permit this tolerance mechanism permits the plant to withstand stress. And acclimation. Acclimation is a non-permanent change in physiology and morphology of the individual to improve response with exposure to environmental stress. So I have worked in the area of heavy metal stress. So I am taking the example of mechanism of heavy metal tolerance in plants. Like first one, I said avoidance. In case of heavy metals, plants prevent the metal transport across the plasma membrane by altering membrane permeability. Secondly, they change the metal cell binding capability also, so that the heavy metal ions cannot bind the cell wall. Third one, they increase the exudation of metal chelating substances, stimulating the efflux pumping. So they start to exudate the metal chelating substances, which chelate the metal toxic metal ions and efflux them from the cell, and there where most of the metabolic processes happen. And one more such process of avoidance is binding heavy metal ions to negatively charged polygluceronic acids in cell wall, so that they cannot enter the cell metabolism. And if this strategy of avoidance fails, like if the quantity of heavy metal ions is very much high, then this strategy does not work, and the plant have to move to the second one, that is the tolerance. So for tolerance, one of the strategy is to cytosolic chelation of the heavy metal ions through different ligands. Suppose the heavy metal ions enter into the cytosol. So plants employ like phytochelatines, metallothioneins. Plant use these ligands to chelate the heavy metal ions and sequester them into the vacuoles. They dump them into the vacuoles, so they cannot interfere with the normal normal cell processes. And second one metals, and third one are amino acid. Organic acid production increases, like proline, histidine, malic acid, citric acid, etc. It increases under abiotic stress, like heavy metal stress. And their possible functions are osmoregulation, protection of enzymes against heavy metal poisoning, and they act as free radical quenchers, which are caused by the stress reactive oxygen species, which are highly toxic to the plant. And one another strategy is vacuolar compartmentalization, like role of VATPs is very important in heavy metal compartmentation, and it is studied that upregulation of transcript level VATP subunit under heavy metal stress. It has been studied that under heavy metal stress, the transcript levels of VATPs subunits upregulates, and the VATPase inhibitors like vancomycin enhances heavy metal toxicity. And another strategy is activation of antioxidative defense system. It is one of the most important defense system plants have or any other kind of abiotic stress that causes the production of reactive oxygen species. Stress, the factors which affect the response of plants against stress are duration of stress, frequency of stress, severity, its combination of stresses, plant character, genotype, tissue in question, and development stage of plant. Plants are more susceptible to abiotic stresses when they are in germinating or seedling growth stage. So these are among the many factors that determine the plant cell response. But even though plants employ so many strategies, but even after these, the biotic stresses are causing damage to our crop production. So we need any alternative to cope with them. So, but that's why the good thing is, in recent years, the advances research of fields of physiology, molecular biology, and genetics have greatly improved our understanding of crop responses to abiotic stresses and the basis of varietal differences in tolerance. So we can apply this knowledge in developing new crop varieties with better stress tolerance and improved crop yield. So, genetic engineering is one such very important technique that offers myriads of applications in improvement of crop plants for both biotic and abiotic stress tolerance. So, genetic engineering consists of isolation of gene of interest. First, we find a gene of interest. We will study them, learn about them in next slides, and then ligating that gene with a desirable vector to form a recombinant DNA molecule, and then transferring that gene into the plant genome to create a new function. And the biggest advantage of this technique is that the desirable genes can be sourced from any source, like plants, animals, microorganisms, or even they can be artificially synthesized. And it has advantages over conventional breeding process. The random mixing of tens of thousands of genes present both in resistant and susceptible plants. So we cannot control which gene will be present in the the plant which we produce after inbreeding. But in case of genetic engineering, we can choose the genes which we want to keep in the product plant. Like it allows the transfer of only the desirable genes to the susceptible plant and the preservation of the valuable economic traits. The resultant plant is called transgenic plant. That is its definition is of uh, relating to or being an organism whose genome has been altered by transfer of a gene from sexually incompatible species. So during last two decades, a large number of transgenic horticulture crops has been developed and more are underway. Plant breeders and geneticists have utilized natural variability for stress tolerance within germplasm. So first of all, like I have said earlier, we have to find the gene of interest. So case of if we want to make a plant resistant to any abiotic or biotic stresses, these are of genes of interest which we want to incorporate in the our crop plants, transgenic crop plants. So first one, genes for osmolyte biosynthesis, genes encoding enzymes for squelching active oxygen species, genes encoding Lia proteins, genes encoding heterologous enzymes with different temperatures, genetic engineer of molecular chaperones, genes encoding transcription factors, and genetic engineering of cell membrane. So first of all, engineering of genes for osmolyte biosynthesis. The tolerance to abiotic stress has mainly been the role of osmotically active solutes like proline, glycine, betaine, sugars such as sucrose, trehalose, fructans, and sugar alcohols like sorbitol, mannitol, etc. are very important to achieve the tolerance at cellular level because they act as osmolytes. So their main function in under stress are to retain water within cell and protect cellular compartments from injury caused by dehydration. Secondly, they maintain turgor pressure during water stress, and they stabilize the structure and function of certain macromolecules, and they act as signaling molecules also, and they also act as quenchers of reactive oxygen species. Next is engineering of genes encoding enzyme for quenching active oxygen species. So we these are also very important abiotic stress resistant because reactive oxygen species cause very much major damage to the plant under stress. So plants suffering from various stresses which lead to overproduction of active oxygen species in cell. To minimize the damaging effect of active oxygen, the genes encoding enzyme with antioxidant capacity such as ascorbate peroxidase, superoxide dismutase, these are genes are of very much interest. And next, engineering of genes encoding Lia proteins. The genes encoding Lia embryo abundance proteins is activated under osmotic stress, mainly under osmotic stress, and play an important role in desiccation tolerance during seed development, maintenance of protein or membrane structure, sequestration of ions, binding of water, and operating as a molecule chaperones. So this is one example given, like the rice plant. When transformed with barley Leah gene HbA1, possessed increased tolerance to water deficient and salinity. And improved salinity and freezing resistance has been observed in yeast transformed with LEAP 2527. Engineering of genes encoding heterologous enzymes with different temperature optima. So, homologous proteins and their specific enzymatic activities from different plant species differ in terms of their temperature. For example, when a gene of NADH hydroxypyruvate reductase from cucumber, which has high optimum growth temperature, it was transferred to tobacco with lower optimum growth temperature. The optimum growth temperature of transgenic tobacco was increased due to transformation with this gene. And next is genetic engineering of molecule chaperones. Chaperone, they assist and maintain correct folding and trafficking of cell proteins. So as we know that protein structure, 3D protein structure is very much important for their normal function. If they do not fold correctly, then they are useless. They cannot perform their usual function. So it is very crucial for plant to survive under abiotic stress so that they can maintain the correct folding and trafficking. So chaperones help in that. So, for example, an antisense inhibition of heat shock protein 70 family prevented Arabidopsis plants from acquiring thermoresistance. And the heat shock protein HSP101 from soybean, corn, wheat, and tobacco complemented a thermotolerance defect in yeast caused by deletion of yeast heat shock protein 10429. And next is engineering of genes encoding transcription factors. So, a response of plants to abiotic stresses are multigenic, and a single gene is not likely to induce the whole cascade. Recently, a transcription factors for abiotic stress-induced gene has been identified, cloned, and used in transgenic experiments. Simultaneous expression of downstream stress-inducible genes have been achieved with parallel increase in stress tolerance. For example, overexpression of transcriptional activator CBF1 induced the expression of 4-COR, cold regulator genes, and increased the freezing tolerance of Arabidopsis plants. And by overexpressing a single stress-inducible transcription factor, lead to plant tolerance to freezing, salinity, and dehydration. And last one is genetic engineering of cell membrane. So cell membrane are very, most important part of any cell. So membranes are critical sites of injury by chilling, freezing, heat, and osmotic stresses. So increased level of if we increase the level of unsaturated lipids in membranes, then it increases its fluidity, which improves the plant chilling tolerance and photosynthetic parameters. So example is a tobacco plant transformed with acyl ACP glycerol 3-phosphate acyl transferase from chilling tolerant species of Arabidopsis acquired chilling tolerance also. So if we employ these genes and we use try to transform our crop plants with these genes of interest, we can very much solve the problem of world hunger and crop production. And therefore it is safe to say that the application of recombinant DNA technology or genetic engineering in crop improvement are immense to solve the problem of global hunger as population is increasing day by day and with depriving sustainable intensification. So that's all from my side. Thank you very much and thank you for listening.
Thank you very much, Dr. Shweta Thakur ma'am. It was a great lecture from your side. Thank you very much for the lecture.
Thank you so much.
Now we would like to conclude our session and end with this international webinar on challenges in global research in agriculture and technology. And I would like to request the organizing secretary of this international webinar, Dr. P.K. Upadhyay sir, as well as the head of the Department of Genetics and Plant Breeding, RVS College, Faculty of Agriculture, Agra, to please deliver the vote of thanks. Over to you, sir.
Heartily pram to all. It was a great experience to see all the invited guests, our specifically invite lead speaker, keynote speaker, and dear delegates who were with us during the webinar both the day. I would like to thank today's lead speaker, Dr. Ulrich Werk from Germany, Dr. Abdul Seok sir from Malaysia, and Professor Rajendra Kumar sir from New Delhi, and Dr. A.K. Rawat from Lucknow, and Dr. Sweta Thakur from Simla. Wonderful presentation today. I would also like to thank Dr. UN Singh Sir, Principal, RBS College, Agra, and Dr. Bipin Agrawal, Campus in Charge, RBS College, Bichpuri Campus, Agra, and all committee members and all faculty of the Agriculture, RBS College, Bichpuri, Agra, and society members and society officials, I.S.G.B.R.D. I also thanks uh, Mr. Anshuman Upadhyay for continuous support in the complete management of the program. Both the day, thank you all for the joining. Uh, thank you and affectionately.
Thank you very much, sir, for your kind words. It was a great session both the days, and this is to inform all the delegates that your feedback form as well as participation certificates will be provided within 1 week, and the feedback link will be sent on your email only. So kindly do not message or mail us again and again for the same. We will send it from our own by tomorrow probably. We'll the feedback links will be sent 100 by 100, as only 100 people can apply for the feedback link one day. So they will be sent accordingly. Rest you have to wait, as the 100 slot will be over, the next 100 will be sent. So within a week we'll complete the target probably, and I wish good luck to all of you, and thank you very much for joining us. Thank you.